Good morning to those of you in the room. Good morning to those of you online. My name is Jim Polli. I'm a faculty member at the University of Maryland and co-director with Aishah from the University of Michigan of the CRCG. And for those of you who are online, kind of wondering what's going on in the room, I'd say the one thing is there's a lot of people in the room. So for those of you who are online, I suspect there's probably about 700 or 800. We have I think maybe about 125 people in the room, which is pretty good for a post-COVID event, actually. So clearly there's a lot of interest in this topic of navigating the transition to low global warming potential propellants. And so this event is, I know some of you are very familiar, so I know you've been to some Center for Research on Complex Generics workshops before. And if you've been here before, then I'll tell you some things you already know. The center was established about 4 or 5 years ago in 2020. It's a collaboration between the FDA, OGD, and the University of Michigan and the University of Maryland. And so the mission is to increase access to safe and effective generic drugs through enhanced infrastructure, communication, education, research collaborations across industry, academia, and FDA. So we really appreciate your being here in person or online. And just to reiterate what the goals of the center are, I guess from Anna and my points of view, as you know, the OGD at FDA has been doing research for quite a long time, and this was started to enhance that, certainly not to replace it. So a lot of our activities are actually more outreach efforts, things like today. We have about four or five workshops a year. So we have infrastructure communication to get the word out on things. We've done a couple surveys, and I think many of you have replied to those, so thank you very much. We published those, and we're on the verge of publishing the next one. We have education and training such as this, and we do have some collaborative research also. Okay, so much thanks for those of you who are willing to talk to us about and answer our questions about what are your pain points and things like this. And so for example, again, we do some surveys, and some of these, I guess, informal communications that we have actually are very helpful in us formulating surveys and then reporting out on those. It's also very informative in terms of even identifying workshop topics and speakers for workshops. Okay, so for this particular workshop, there's a packed 2-day agenda, a lot of experts that will be speaking, and we appreciate all of their willingness to come in and share their insights. In some ways, we've sort of been through this before, right? So hopefully lessons from the prior transition from CFCs to HFAs will bear out. And a colleague of mine at the University of Maryland is Richard Dalby. Those of you who've been to RDD, I think, are very familiar with Richard. And he told me that there's been an application that was submitted earlier last week involving a reformulation and a novel propellant. And I'm sure later today we'll maybe hear more about current activities in that area. So just a little bit more about the CRCG. We've been around for a few years. We've had 16 workshops and training to date. We tend to have about 1,000 people online, and maybe, I see Richard there. Hey, Richard. We tend to have about 100 people in the room. And just in terms of advertising for 2025, we have two workshops in the spring on in the transdermal area, IVPT, and also on particle sizing. Those are actually have a hands-on component also, a little bit of demonstration about those techniques. And then next fall, we have one on modeling and artificial intelligence in terms of generic drug development, as well as one on ophthalmics. We do have some research on the go, and over the years, here's some research that we've been involved in. For example, some of our research is, this was highlighted a little bit at the last CRCG meeting last month on the nitrosamine topic. Okay, again, my name is James Polli, and Aishah is here. Visha's online, Dana's online, and Jennifer's out front. I think you've probably interacted with Visha and Dana and Jennifer before, maybe. And please sign up. We have a website, complexgenerics.org. If you want to get more information periodically, we try not to send too many emails out, but that's how we do that. So you can sign up if you want to be on our emailing list. Okay, much thanks to the FDA Planning Committee, in particular Elizabeth and Brian, along with many other people. And then we also have, because it's a CRCG event, we also want, we actively want industry to be engaged and have their perspective, and much thanks for the group in doing that. Okay, so thanks for your participation. Just one comment, in about 3 weeks, probably before Christmas, we'll be able to provide all these materials online. So the videos are being recorded, and speaker handouts will be available as PDFs, and we will just email that. If you register for the event, you will automatically get that, that notification. Okay, so it's my pleasure to introduce Markham Luke, but to provide some introductory comments, but before I do that, I would just want to introduce Sally Seymour, who will be moderating the first session. So Sally is the Director of Pulmonary Allergy and Critical Care and OND. She has a MD degree from the University of Cincinnati, although this audience may be interested in knowing she also has a BS in Chemical Engineering. So she's been in pulmonary allergy and critical care for a couple decades. She's very familiar with pulmonary products, things like metered-dose inhalers, and she was there during the transition to HFAs. And then also Markham. I think many of you know Markham. He's been around a while. He's a supervisory physician in the Division of Therapeutic Products 1. He has his MD and PhD degrees from Hopkins. DTP1 is responsible for pre-application development in generics with an emphasis on research. You may know him in many different ways. He's a dermatologist. He was the Chief Medical Officer in the Office of Device Evaluations, and he was even Acting Director of Cosmetics. Okay, Markham.
Thank you, Jeff. Thanks for all of the folks who are joining us this morning. Division of Therapeutic Performance and Office of Research and Standards in the Office of Generic Drugs. We have a lot of folks here in the room, a lot of folks online, and we're going to have a great meeting to discuss navigating the transition to low global warming potential propellants, the LGWP propellants. And make sure you put those 2 P's at the end of it. And so onto our topic. The disclaimers, this presentation is not to be construed to represent FDA's official views of policies, any representation of brand name drugs, and just talk for scientific discussion and specific comparison. And there's a little picture of a boat navigating stormy waters. So to introduce the topic, complex drug product dosage forms include metered-dose inhalers, and metered-dose inhalers are going to be the main focus of our discussion. However, the propellant switch does affect other drug products as well. And so there'll be folks keeping an eye on this space and how we deal with this. These products include other spray products with propellants that are regulated by CDER and FDA as well. So these inhalers date back to the 1950s, during that first decade after World War I. When they were looking for a safe and effective method to deliver metered-dose inhalers. And these have been in our product pipeline for over 70 years. And they account for about 2/3 of all respiratory products prescribed. The main targets are asthma and COPD. These products are formulated to deliver a range of active pharmaceutical ingredients, including corticosteroids and short and long-acting bronchodilators. Those are the main ingredients that we see in these products. They're intended to provide a fast and convenient aerosolization system for delivering the drug, and they do require propellant to administer the drug. These metered-dose inhalant propellants perform multiple functions. They are the driving force. They're physical action for pushing the drug into from the actuated device into the lungs. They facilitate atomization of the formulation into aerosolized droplets, and these droplets then can be deposited in their forum to be adsorbed into the lung tissue, et cetera. They constitute the bulk of the MDI formulation, providing solubilization for dissolving and dispersing the API in the formulation. So some history on the MDIs, and I know there'll be talks during our first session that Sally's planned to discuss a lot more about some of the history and ups and downs in the progress of these metered-dose inhalers, with a specific focus on the first generation, the CFCs. And those came out years ago, but then they found out that this affected the ozone layer. And so there was a 1987 Montreal Protocol, and then during the 1990s, there was a phase out of those products, leading to second generation products with HFAs, which don't deplete the ozone layer, but they do have been found to cause potential for global warming. And they've been used in all current metered-dose inhalers, and that leads us to the current phase down for the third generation. And this is what we're talking about today. And by 2047 is the target goal for this. And we have folks from the EPA here today as well. They're going to talk a little bit more about some of the reasons why we're doing this, and what the bigger picture is for the phase down for these propellants. There is a potential for shortages of these products and cost increases for the raw materials going into products that utilize these propellants. So manufacturers will be driven to look for alternative propellants for the metered-dose inhalers. But along with that comes the scientific question of how do these the new propellants perform in the context of the products that we're regulating. The removal of these CFC propellants from the market is a complex multi-year process. Back in 2005, they started in, like I mentioned, in the 1987 Calgary Montreal Protocol discussions, and then that throughout the 90s and then into 2000, there was a discussion. So it took multiple years, and this is important to keep in mind that it was a gradual process, and to remove the CFCs, each metered-dose inhaler required reformulation. The HFAs, which include HFCs, became the new propellants. And due to different chemical properties between the CFCs and HFAs, there was actual redesign of the device that was sometimes necessary in addition to reformulation. And so new formulated MDIs require full clinical development programs, and there were also patents and exclusivity being listed for the new propellants in the Orange Book. And this, of course, led to some problems with generic adoption and the phase out for these products. Here's a timeline, and I think my slides will be available. I'm not going to go into detail on this, and there'll be additional discussion of the timelines. But if you want to look at the timelines closer, they'll be available on the CRCG website. The key thing here is that there were the phase out from the market for these CFC propellants impacted generic MDIs in two ways. They removed the existing generic MDIs in the marketplace, and then the patents and exclusivity for the HFA propellants contribute to delays for the generic versions. And this is a concern, and we're trying to see what we can do to streamline our approach for the new phase out or phase down. So this CFC phase out had a broad impact, and this will be discussed further by Dr. Seymour during her first session, so stay tuned for that. There's a couple of graphs here on the sales of the brand name albuterol inhalers from the late 90s, from the 1990s to 2021, and you can see there was a area there was a lack of availability for some of these inhalers, and that also led to delays in getting generic products out for the albuterol inhalers. And so this Key thing for the next propellant transition that we're all here today to learn more about is the discussion that we've been having with EPA, et cetera, on this phase down. So these HFCs contribute to climate change, while HFC emissions from MDIs represent only less than 0.1% of the total greenhouse emissions. The efforts still are underway to address their environmental impacts. The Kigali Amendment to the Montreal Protocol in 2019 said we need to phase down the consumption and production of all HFCs. And so that's the global issue, that these HFCs are going to become less and less available. And countries that are allowed exemptions for certain essential uses, there is a 2020 American Innovation Manufacturing Act that directs EPA to address this phase down of HFC by law. So EPA has published a final rule for the phase down of HFCs that mandates a phase down for HFCs to 15% of the baseline levels over a period ending in 2036. We've been looking at applications, and companies have been trying to get application-specific allowances through next year. 2023, the final rule for HFC allocation was published by EPA, and they continue to be looking at how allocation is going to work beyond 2025. And the EPA is here, and they may be discussing more about that today, this morning. Communication between EPA and FDA is ongoing to minimize the effects of these HFC phase down in the immediate dose inhaler market. Prevention for any potential for drug shortages due to propellant transition is important. HFC allocation for MDI drug developers, along with drug development timelines for completing propellant transition, have been discussed. And you can see on the side there, the graph about the phase down and anticipated ongoing lack of availability for the HFC that you can anticipate through the upcoming years. Implications for global HFC phase down. Countries around the world are working to phase down the HFC use. So we're mindful of that, and day 2 we'll have an international session where some of our international regulators will be joining us as well to talk about what their plan is for phasing down, and we can hopefully get a global coordination on this effort. I think some of them are going to be joining us online, so through the benefits of modern virtual technology. The differences in phase-down timelines and regulatory requirements across health agencies could lead to some challenges for both brand and generic developers. There's coordination and communication amongst the health agencies for this important transition to make it most efficient as possible. We have a generic drug cluster, which we'll talk a little bit more about in day 2 of our meeting here. The health agencies begin to provide guidance in 2023. EMA published scientific guidance on Q&A guidance. And if you read through it, it's actually a good read. There are some studies that they recommend that we're looking into seeing are these scientifically valid, and are these potentially approachable. It's not entirely clear when you look through the EMA guidance document. Importantly, this transition is a phase down of HFCs, not a phase out as previously done with CFC propellants. Therefore, this time the HFA MDIs can remain on the market, including generics. However, as the phase down of the HFCs is occurring globally, there's an expectation that this is going to impact both the supply and cost of HFAs. The increase in cost of HFA propellants is likely to impact manufacturing decisions for HFA-based MDIs that could increase the cost of these treatments for patients, and we're trying to minimize that as much as possible. The other thing is that these manufacturing decisions, we know, need to be made way in advance because you actually have to change your manufacturing plant. There's a lot of issues with logistics that impact how the products are manufactured with the new propellants. You can't just plug in a new propellant canister into the factory assembly line and expect it to work. There's a lot of discussion about how manufacturing plants might need to be refurbished, or even new plants being made. So ideally, this current phase-down, phase-in propellant change is conducted in an orderly fashion, maintaining access to these important life-saving disease-treating medications for our patients in the United States, which is our focus at the FDA. We strive for balance with regards to least burdensome approach, but yielding products that are therapeutically equivalent to the ones they're replacing. And with that, I wanted to thank Dr. Brian Newman, who has put a lot of work into thinking, along with the rest of the team, about what things need to go into this session. Rob Leidenberg, who's my boss, who was one of the first to mention in one of our workshops that, hey, this transition is coming back in 2019. We need to be prepared for this. And so we were given plenty of lead time to have these discussions. But the key thing is to bring other parts of the agency, other parts of industry and academia on board so that we can work together to facilitate this transition. I want to thank our FDA CEDAR Lifecycle Management Board and our O&D OPQ colleagues for helpful ongoing discussion about the paths forward. We continue to embrace and we love working with other parts of FDA to enable both this change in complex product regulation as well as others, and our EPA and academic industry collaborators as well. So thank you. And with that, I'm going to, Sally, you're next. I think we're a little bit early, but we're on time. With that, everybody can take up a chance to stand up and stretch maybe, and then welcome Sally to the podium. Thank you.
Okay, good morning, everyone. Welcome to session 1 of the workshop. My name is Dr. Sally Seymour, and I'm the director for the Division of Pulmonology, Allergy, and Critical Care in the Office of Immunology and Inflammation, in the Office of New Drugs, in the Center for Drug Evaluation and Research at the FDA. So that's a long list of organizations. We'll just shorten it to this morning's session is going to be focusing on the transition for new drug product metered dose inhalers. And I'm going to be introducing the speakers. I'm also the session lead and the moderator for our panel that follows our presentations. You have a QR code on your table that has the bios for the individual speakers. So I'm not going to be going over the bios in detail. If you want more information on the individual speakers, you can use that link to get more information. Listed here are the speakers and panelists for session 1. And you can see that we've organized this to provide a little bit of foundational information for this transition. So our first speaker is going to be from EPA, Luke Hall-Jordan, and he's going to provide some background about the HFA phase down. I'm going to come back and give a very brief presentation that overlaps a little bit with what Markham talked about this. And then we're very grateful to have Stein provide some of the basic information about the new propellants that are being considered and some of the considerations that manufacturers and developers need to think about as they're reformulating. The next group of speakers are all from FDA, and they represent a multidisciplinary sort of considerations that you need to think about and what our current thinking is for the transition. And again, this is for the new drug products. And so I'll introduce each of them before they come up. All of the presenters will also be on the panel discussion. I'll be moderating that. And then we have an additional panelist, Zhang, who is from OPQ, the Office of Product Quality at the FDA, and she's the director of DPQA 12. And she's an expert in biopharmaceutics. So she's going to be joining us as a panelist. Just a couple housekeeping things for how this will run. We are not going to be taking questions during the presentations or after the presentations. So the questions, you can save your questions and we'll take them during the panel discussion. So please hold those for that. I think those are all the housekeeping things we need to talk about. So with that in mind, I can introduce the first speaker. Luke is a supervisory environmental policy analyst in the phase down implement branch at the EPA. And thank you, Luke, for joining us this morning.
All right, good morning. Nice to see you all this morning. As Sally said, I'm Luke Holt-Jordan. I work at EPA. Our group is focused on the phase down of ozone depleting substances as well as hydrofluorocarbons. So we also have a little bit of that institutional memory of the transition for MDIs from CFCs back in, started in the 90s. So I'm gonna walk through, give you a little more background, dig in a little further. I know Markham talked a bit about sort of some of the overarching transition and information on some of the regulatory and international space, but I'll dig a little bit deeper. Okay. All right. So, yeah, skip that. All right. So first, I think, we want to sort of provide out or face down a class of chemicals that's relevant for MDIs. Back the Montreal Protocol was originally agreed to in 1987, and that led to domestic regulatory action in the United States under the Clean Air Act to phase out. Just want to emphasize that distinction: phase out CFCs with limited exceptions. EPA stood up regulations to phase down, focusing primarily on the production and import of the chemical itself. There also are some use restrictions and other things that we put in place to phase these chemicals down. Now for MDIs, oh, that's better. I assume that's a little louder. Yeah, okay. For MDIs, there were exemptions under what we called the Essential Use Program. The process was, was a sort of had domestic and international context that drove how that would work. So it was a 3-year cycle where manufacturers would come forward. They would talk to us and give us information on how much CFC they would need for the products that they were making, the MDIs that they were making. EPA would work very closely with our colleagues at FDA and Department of State to put together a U.S. nomination that we would put forward to the Montreal Protocol, this international treaty that governs the global phase out of ozone depleting substances. Long story short, we would go to the parties, we would, work with the other governments that were there, we would explain our need, we'd advocate for those needs, and then the parties would make a decision on a recommended amount of additional production that was allowed, and then we would take that information, bring it back to the United States, and allocate what we would call allowances for production of the amount that was needed. Now, that was a 3-year cycle, so we were continuously working to try and figure out into the future what it would look like. I provide this context just because it was an arduous process. It involved a lot of work, a lot of coordination, involved an international sort of review. So Markham mentioned sort of the Montreal Protocol. Up until the Kigali Amendment to the Montreal Protocol in 2016, HFCs were not part of that international treaty. There were no restrictions. Obviously, every country had different controls in place. I know we had some domestically, other governments, I know the folks in Europe were further ahead in their phase down schedule for HFCs. But what this did was establish a series of schedules for different countries globally to phase down primarily the production and import of the chemical itself. That's usually where the protocol is focused, is on the actual chemical. It's our schedule, has us stopping at 15. Different from the CFC, which we had, phasing out other ozone depleting substances. It's really hard for some applications. Transitions can take a lot longer than you expect for some of these. And so recognizing that not all applications would be able to transition, potentially, the phase down schedule goes to 15% of baseline. What this means, right, is that certain uses like MDIs or others could use HFCs unabated long term, depending on what the rest of the the market for HFCs is doing. So to put into context the benefits, right? So what are HFCs? I think you all usually refer to them as HFAs for the two propellants that are used in MDIs. They were introduced, they're man-made chemicals introduced as replacements for ozone depletion. By 2100. So that's really significant. I think if you follow any of the international climate, the goal is to keep warming between like the 1.5 degree to 2 degrees centigrade. And if we're talking about from just this class of chemicals globally, you're looking at 0.5 degree. That's pretty significant. A pretty significant contributor there to help keep that number lower. For folks that may not be as familiar with global warming potential, just what it is, because it is a term of art, it's meant to allow for comparisons between different gases. So it basically is a measure of how much energy the emission of 1 ton of a gas will absorb, how much energy does it pull into the system and lead to climate change, relative to 1 ton of carbon dioxide. So it's just sort of a scaling feature that helps us sort of compare across different chemicals and what they would mean. Usually there's a time horizon that we're looking at. Usually it's 100 years, so that gives you a sense of when it's emitted over a 100-year period. Okay. Great, so I'll pivot a little bit here to the domestic side. So in 2020, Congress passed and the President signed the AIM Act, or the American Innovation and Manufacturing Act. It's similar in many ways to what we did under the Clean Air Act to phase out ozone-depleting substances, with some important differences, but it sort of falls into three bins. We often call it like our three-legged stool, where you have 3 different programs that are helping to support this phase down. The first is really on the supply of the chemical. That's actually what my group focuses on, as we focus on the production, the import of the chemical. and implementing an allowance allocation and trading program, which gives people these, they function like permits that allow folks to import or produce a certain quantity of chemical. So that's first one, supply, reduce the supply of these chemicals over time. You also have a group that focuses on, well, what are the alternatives people can transition to? Are those, reviewing those, making sure they are, relatively safe and effective compared to the one that's being used today? And then also looking out ahead to say when there are alternatives that are available for, say, a high GWP HFC, maybe removing the ability to use that high GWP HFC to make more space within that allowed supply for uses that are harder to transition. And then our third approach, or third leg of the stool, is managing use and reuse of HFCs. So, it's made, it's used in a product, and it's going to be out there in the economy for years. You still have equipment that uses chlorofluorocarbons as the refrigerant, now 30 years old or more. So, it's making sure that the use is happening properly, make sure we're recycling it, make sure we're reducing emissions. Again, to help ensure there is supply of that chemical to support systems and equipment that's still using it. So that's sort of the way that Congress sort of structured our domestic authority for phasing down HFCs. All right, so you've seen this step down schedule here. Right out of the get-go, we were at a 10% reduction relative to the historic baseline levels. And by 2036, we'll be down to 15%. That's the schedule we are directed to follow. This year actually was the second step where we dropped from 90% down to 60% of the historic baseline level. So already starting to decrease how much HFC is available. I will note that the way the schedule works, it's not about the actual quantity of HFC that's produced. It's about the GWP weighted quantity. So if you switch from a higher GWP HFC to a lower GW HFC, you could potentially produce the exact same amount or more, at a lower environmental impact. So I think what we are expecting is that as this transition plays out, folks will be transitioning from high GWP HFCs to low GWP HFCs and non-HFCs. So there'll be a mix. And I think, again, you'll note that it's a phase down, it's not a phase out. It is possible for certain uses to continue using HFCs, unabated. There could be implications, which folks have mentioned already, Markham mentioned already, about supply and cost. But in terms of what can be produced, there is space for different decision making. All right. So this will be in the slides. I'm not going to go into this in too much detail, but just to note, we do have a number of regulations that are in place for all three legs of the stool. The most recent of which was just back in October, where we finalized our final sort of framework rule that would establish the emissions reduction and reclamation program. The other thing I would note is for MDI specifically, they are not covered in that emissions reduction and reclamation final rule in that second column. They are not covered by the technology transition requirements at this point in time. So really, when you're looking at our regulations, it's really in that first bin of the regulations focusing on supply. And I'll talk a bit more about that program. Okay. So under this HFC allocation program, we call it for shorthand, this started we finished a rule in late 2021. Congress gave us the leisurely schedule of 270 days to develop a whole new regulatory scheme, which for folks who are familiar with the regulatory process is really fast. But we did set up a program that would allocate allowances, which again, permit people to produce or import these HFCs. And there's sort of two parts of, the allocation. There's what we would call the general pool of allowances, which goes to folks who are historically producing or importing HFCs. Usually that's the chemical manufacturers or some of the other folks that are importing, from abroad. You also have what we called application-specific allowances, which is particularly relevant for MDIs. So I'll talk about that in a bit. That initial rule, it set up sort of the framework that we were going to operate under. It established the application-specific allowances and eligibility for 5 years through 2025 for 6 applications, including MDIs. It also, established how we would allocate allowances to the general pool allowance holders, those historic producers and importers. Now, allowances are issued every year. We have to issue up to the cap, the line, on the top of that line that was on that bar chart on the last slide, or that graph on the last slide. We since have finalized a rule last year that would go through 2028 for the general pool. And again, we're going to have like 2 rulemakings basically going on about every 5-year cycle to do general pool and application specific going forward. So let's talk about application specific, because that's probably the most relevant for this meeting. Some of you may be very familiar with these requirements, so forgive me for going through them again. But when Congress passed the AIM Act, they wanted us, or directed us, to give priority access to HFCs for certain applications. And they listed 6. And metered-dose inhalers, use as a propellant in metered-dose inhalers, is one of those 6 applications. And so the agency did set up what we've called this application-specific allowance program. So instead of providing the allowances to the producer or the importer, the supplier of the chemical, right, the allowances are provided to the user of the chemical in that application. So in this case, it's the companies that are manufacturing MDIs. The way that the allowances are intended to work is, we will allocate them to the user, and then they can work with their supplier however they want to confer the allowances and authorize production or import on their behalf. Now, in this process, we work very closely with our colleagues at FDA when we're talking about trends that are emerging, when we're talking about different allocation decisions, and just trying to monitor what might be coming that we're not aware of, because surprises can always be challenging for anybody. And obviously, I've been appreciating that partnership. I think that's one thing you'll note throughout the 30-plus year history of our work to phase down ODS and now HFCs, is that there's always been that strong collaboration. All right. To give you a snapshot of what we're basing allowance decisions on each year, we have annually, companies have to request how much they think they will need for the next year. We're basing then the amount that we can allocate to them on, how much have you purchased or used over the past 3 years, and we're multiplying that by their growth rate, average annual growth rate over that time horizon. We also recognize there are certain situations that just, they may not be covered by that formula. So we have what we call these unique circumstances or opportunities for folks to request additional allowances above and beyond what they might have historically purchased. And that could be, an additional manufacturing capacity coming online, which we've also interpreted to mean new products are coming to market. It could also be acquisition of another manufacturer, its facilities. I don't see it happen as often for MDIs, but no, for other applications, they are often buying different facilities. And this gives us a way to ensure they have the allowances that they need. And then the third is very specific to MDIs, but pandemic or other public health emergency that increases patients treated using MDIs. So I think this is one where, again, if there is something that's driving additional demand for MDI, sort of that patient-centered approach, then this gives us an opportunity to consider that as an opportunity to give more. You can see where the pie chart here shows application-specific allowances are a relatively small piece of the overall allocation, about 5%, but not insignificant. All right. There are reporting requirements. If you have to report, you probably know what they are. I'm happy to talk with you further, and they're here in the slides if you want them for later. But we use this data to determine how many allowances a company would get, and also sort of track trends in terms of where things are headed. All right, so let's talk about 2025. I mentioned it was about 5% of total allowances. This pie chart here just shows that pool of application-specific allowances. MDIs is a little under 15%, similar to the blue, which is actually semiconductor use. Green is the light green is that mission-critical military end uses, so the Department of Defense for their needs. We also have defense sprays, certain foams that are used in trailers and marine or boats, basically, and then onboard airspace fire suppression. So these are the sort of 6 applications, that's where they fit. And I've already highlighted the fact that these allowances are a little bit different in the sense that they're allocated to the user and not the chemical supplier. Okay. All right. So just to note, I think EPA has to go through a process every 5 years to review whether to renew eligibility for access to these allowances for users like MDI manufacturers and others. We are in that cycle right now. We issued a proposed rule earlier this year. And consistent with the statute and consistent with the AIM Act, right? We can only renew them for up to 5 years, and then we've got to go back in and do another assessment. So this rule looks at 2026 through 2030. And we did propose that MDIs would be renewed for the full 5 years to receive application-specific allowances. We also, based on our experience, we also put in proposed some additional flexibilities. One of them was related to sort of healthcare system needs. Again, it's not the same as a public health emergency or a pandemic, but there's some sort of unexpected healthcare system event that causes, a change in what an individual company might need. I personally think this will be quite relevant for the transition, where you do have folks, maybe stopping producing their branded MDI, and we may have generics coming in to replace it. So that could cause some shifts that may not be reflected by what the standard formula would be for how we would allocate. But again, we've taken comments on that. We're reviewing those comments and working toward a final rulemaking in the summer of next year to issue allowances for the next 5, or really to set up the formula so that we can issue allowances for the next 5 years. If you have questions, obviously feel free to follow up. There were some other sort of minor tweaks in there related to the reporting requirements, the methodology for how we would allocate allowances, but are all sort of based on our experience over the past few years implementing the program. And hopefully, again, create that flexibility where it's needed and in a justifiable way so that we're able to allocate what companies need for these covered applications. All right, so that's what I've got. There are some resources here on our website. There's a wealth of information. And I've also, when you do get the slides, you have some contact information to get a hold of us if you have any additional questions. So thank you.
Okay, I'm back. I'm going to give a very brief presentation on lessons learned from the CFC transition. This overlaps a little bit with some of Markham's slides. You could probably spend a whole session talking about lessons learned from the CFC transition. And as many of you know, it took about 20 plus years. So I'm going to cover that in 10 minutes. So it's going to be high level. This is the standard disclaimer slide you're going to see from all of the folks from FDA. I won't go through it, but we don't have any conflicts of interest here too. All right, just a slide on the history of metered dose inhalers. Really, the main thing I wanted to say is, these have been around for a while. The first metered dose inhalers used CFCs as the propellants. You know a lot about the CFCs as you guys are manufacturing and developing these products, but they had a wide variety of applications, and one of the things that they were used for, in addition to air conditioning, refrigeration, et cetera, was for being propellants for MDIs and other commercial products. But they were also used for cleaning metal, which I think we came to appreciate during the transition when some of the newer products had a lot of issues with clogging. So I think the CFCs were probably propellants that help keep the MDI clean, and that's something that we learned during the transition. The CFCs 11, 12, and 114 were the propellants that were used in MDIs until they were phased out. So just a little bit here about the background. I mean, this has already been covered some from Luke, but probably the article that's mentioned most is this 1974 Nature publication from Molina and Rowland that linked ozone depletion to stratospheric chlorine from degraded CFCs. And I think this helped with EPA publishing rules regarding the phase out of CFCs. But they did have some clause for essential use exceptions, which did include metered dose inhalers. The worldwide community came together on the Montreal Protocol on substances that deplete the ozone layer, and the US signed that in 1987. And as Luke mentioned, this was a worldwide treaty to actually phase out the use of CFCs. And in the US, in 1990, the Clean Air Act was amended to codify our commitment to the Montreal Protocol. And in 1996, the CFCs were phased out in industrial countries, except the essential medical use, which included MDIs. And this was a multi-cross-agency effort. This included work with the State Department, who represented the US on the Montreal Protocol. The EPA, obviously, we worked closely with them on this phase out. And then the FDA was actually sort of tasked with how to phase out the use of CFCs and the MDIs. So from our perspective, we did this process through rulemaking. And 21 CFR 2.125 is the overarching rule that was used to prohibit the use of CFCs in FDA-regulated products. But they did have some essential use exemptions, and this was for MDIs, which meant that if you had an essential use, you could still use CFCs for the production and manufacture of those products. And this regulation went by individual moieties, like albuterol, and went through the process of what was essential use for the individual moieties to allow them to continue use of the CFCs. So we had to determine criteria in the framework for when that essential use was no longer needed. And so that was a complex process. It involved public input either through advisory committee meetings or open public hearings. We published proposed rules in which there was feedback from stakeholder and comments. These proposed rules would propose end dates for when the essential use would no longer be there. And then based upon the feedback and all this process, we published a final rule to remove the essential use end date. So a complicated process that was resource intensive. And in addition, we had to work with the EPA on the CFC allocation process, and also the companies on their reformulation programs, and then review the applications that came in. So this is a complicated process that, maybe with this phase out, we can streamline and not have it be so burdensome. Phase down. I'm not going to go through this timeline in detail. I went through the yellow steps already that are some of the legislation or the treaties that we signed. Just to note, though, that there are a number of advisory committee meetings. We first had to develop the framework for what the essential use framework was going to be to remove that, and then a couple advisory committees to discuss specific moieties that had sort of unique considerations like albuterol because of its widespread use and the impact on patients, and epinephrine because that was used over the counter. And then the red boxes show the collection of moieties and their essential use end date. And after that date, they can no longer be marketed in the US or manufactured in the US for sale. So you can see that this process took a couple of decades to go through. Now, since I'm from FDA, I mean, I provided some FDA perspective, but obviously this had major impact on other stakeholders, industry in particular as well. The HFA propellants were quite different than the CFCs, and so there was significant research and development necessary to identify these new candidate propellants and determine reformulation was necessary because they were so different. The surfactants and materials, there were compatibility issues, device changes, and extensive safety data had to be generated to support the use of these in MDIs. New manufacturing processes and complete new development programs, submission of an NDA and review, and estimates that I found in one article were between $250 to $400 million of investment. So a lot of time, research, money was spent by pharma on developing the new MDIs. And so companies had to make a decision. We were either going to reformulate the MDI and use the new HFAs, or we're going to change platforms. And a number of companies did change platforms and develop DPIs or soft mist inhalers that don't use any propellants. But some companies decided to just get out of the MDI market because they couldn't make the case financially, business-wise to reformulate. And so we did see products fall away from the market. I do think that the CFC phase-out did encourage some innovation in drug delivery. Now, this is a poster from current available platforms, but we did see more interest in DPIs and also, as I mentioned, the soft mist inhaler. So I do think this was maybe one good thing that came out of the CFC transition is some innovation in drug delivery. So there was a significant impact on the CFC phase-out in generic MDIs. In 1995, the first generic CFC albuterol was approved, and by 1997, we had 4 available in the US. However, from the timeline, if you remember, in 2009, the essential use for albuterol was removed. And this impacted not only the brand products, but also the generics. So none of those could no longer be manufactured. So essentially, after 2009, only the HFA albuterol MDIs were available, and these were all branded products with branded prices. And in 2020 is when our first generic albuterol HFA MDI was approved. So we had over a decade time for patients where they didn't have a generic alternative for albuterol to purchase. So this is a significant impact on generic drug. So, you know, our ultimate customer here is the patient, right? This is what we're all here for. And so what's their perspective on the CFC phase out? I think that they had some challenges when they got these new MDIs. They looked the same, the user interface was similar, they kind of knew how to use them, but for them, the experience was very different. And we heard about this and we heard complaints, and I'm sure you did too as companies. Particularly this softer plume. Patients just had the perspective that if I don't feel that force at the back of my throat, this isn't working for me. They also had to actually prime and clean their inhalers, which they really probably didn't have to do with the CFCs. But in order to keep these functioning, this was something new for them. Some patients also saw the discontinuation of products that they relied on and used for years and were attached to a certain particular MDI. And I know that that was difficult for patients as well if manufacturers didn't reformulate. I mentioned the generic albuterol was no longer available. So this was a huge impact for patients financially. I mean, it was a big increase in prices. So I think for patients, I don't know if they knew this was coming for their inhalers, but they were definitely impacted, and it was a challenge to really, I think, educate consumers on the difference in these inhalers. Markham showed this slide earlier. Really, this is to show the sales of some of the branded products. This is for the albuterol. A couple key things here to note is that initially there were only branded albuterol MDIs available, but by 1997, when you start to see a decline in the, sales here, that's when the generic MDIs for albuterol were first available. And for patients, they may have been paying around $15 or less for those. And by 2000, in this area here is when we had about 4 generic albuterols available, and you can see that the sales for the branded products were declining, probably because of the generic competition. But 2009 is when the CFC phase out for albuterol happened, and you can see that's when the HFA sales for the albuterol products started to increase. But also, that's when the out-of-pocket expenses for patients increased to over tripled, and I've seen some estimates of over $200 for their monthly albuterol MDI. In 2022, now we have actually about 4 MDIs, HFA albuterol MDIs, that are proven available, and the cost is coming down for consumers to less than $25. So significant impact for the change in propellant from the HFAs, or CFCs to HFAs. Okay, to sum up in a couple slides here, the CFC to HFA transition was a very complex multi-year process. There is a major impact on formulation in the device that required reformulation to MDIs, to new propellants, or develop another platform. This was very complicated and burdensome for us as well as industry in terms of rulemaking and advisory committee meetings, et cetera. There were full clinical development programs for the new MDIs, which were costly for industry as well as downstream effects on patients. As Markham mentioned as well, these new HFA MDIs did have patents and exclusivity attached to them, which prevented HFA generic product development, and all of this led to an increased cost of the new HFA products for consumers. So looking forward for this phase down, this is not required in terms of an HFA phase out. This is a phase down. And approved brand products and generic HFA products are not required to reformulate. However, we do understand that there are going to be potential supply chain and cost issues, so many sponsors will develop MDIs with these new propellants. And from what we've seen so far, we're still early in this transition, that the properties of the HFA and the low global warming potential propellants are more similar than they were to CFCs. So we're looking for opportunities to streamline these development programs, and our presentations from FDA today will explain some of our current thinking about when you can streamline and what that streamline program might look like. And we're really leveraging and considering their experience and knowledge gained from the generic MDI development programs and looking at their generic product-specific guidances. So you'll hear our perspective here in the next set of presentations after Dr. Stein presents, but we're really, as Markham indicated, looking for a smooth and orderly transition and really trying to think about minimizing the impact on patients as well as the impact on generics and costs. So with that, I will close my presentation and I will go ahead and introduce the next speaker. Steven Stein. Okay, I haven't met you yet, so it's nice to see you. I'm glad you're here. Steven is the scientific director of inhalation product development and Condiva drug delivery, and we appreciate you being here today to talk to us and give us a basic sort of understanding of the propellants that we're looking at for reformulation and their impact on product development. So thank you.
Thanks to the organizers for the opportunity to present here today. So my task is to provide a technical perspective on the impact of the propellant change on product development and product performance. The transition from current to LGWP propellants is well underway. As it has been mentioned, the differences in the properties of the propellants for this transition are not as significant in terms of chemical properties. The changes are not as significant as the CFC to HFA transition, and yet there are important differences in the properties that impact product performance and formulation considerations. And so we'll look at some of those in this presentation. Here's an outline of my talk. I'm going to start by just talking about some of the processes occurring during MDI atomization and dose delivery, and then we'll look at how the propellant can impact those processes and the impact on key metrics like APSD. We'll also look at the impact of propellant on drug and excipient solubility, suspension behavior, and spray pattern. All right, so turn the technical part of your brain on for a bit here. When an MDI is atomized or actuated, there's a lot of interesting physics that happens in the actuator as the propellant passes through the valve into the actuator sump and out of the spray orifice. Now, these are some measurements of the pressure inside the actuator sump during atomization for the different propellants. And within a few milliseconds, the pressure in the expansion chamber increases significantly, up to about 4 to 5.5 bar, depending on the propellant. That's right near the vapor pressures of the different propellants. You have an increase, but then as the valve begins to empty, that pressure diminishes. And this whole event is a complex event that happens in a few hundred milliseconds. I find it helpful to try to break down the MDI dose delivery into two phases. The first phase being the atomization phase, where the formulation is broken up into small aerosol droplets, and then the second phase where the droplets evaporate and you have the plume interacting with the patient as they take a dose. Now, this is a real interesting image here, interesting X-ray image inside an actuator sump during dose delivery. And what you see is a complex multi-phase flow. The lighter regions represent propellant vapor bubbles. The darker regions are the liquid propellant. And because of the high vapor pressure, that mixture is forced out the spray orifice. And when that happens, the liquid portion of the formulation is stretched into ligaments that break up into small droplets. Now, the size of the atomized droplets depends on the vapor pressure, the surface tension and viscosity of the fluid. So a lot occurring during that atomization phase. Then when the droplets exit the orifice, they come out at a high velocity, sometimes up to 30 to 60 meters per second. So you have a high velocity plume that interacts with a much lower velocity airflow in the patient's mouth and throat from their inhalation. So you end up with a lot of turbulence that occurs in that region. The velocity of the particles are rapidly decreasing. The size of the droplets are rapidly decreasing as the propellant evaporates away. And you get some deposition, impaction, or turbulent deposition in the mouth and throat. Now, what are some of the properties that impact the atomization and evaporation processes? Well, okay, first off, why are we here is because of the bottom line. I'm going to let others talk about that as they already have. But in terms of what impacts the dose delivery, as I mentioned, the vapor pressure, surface tension, and viscosity, they play a role in that they impact the size of the atomized droplets, which can impact dose delivery. The latent heat of vaporization and the vapor pressure, those both impact how rapidly those droplets will evaporate away. And I'll talk a little bit about that later. In terms of formulation factors, the densities of the propellant is an important difference between the propellants that can impact the formulation in a couple of ways. The first way it impacts the formulation is that if you're trying to match the dose of an existing product using one of the current propellants with one of the new propellants, because the densities are lower for the new propellants, if you want to achieve the same, deliver the same dose for the drug, you need to either increase the concentration of drug in the formulation or you need to increase the valve size. So that will impact the design of your product. In addition, the density is important in that the settling and creaming behaviors of the propellants can be quite different. The dipole moment can impact the solubility of drugs in the propellant. And there are some differences between the propellants. But for context, the CFC propellants had much lower dipole moment, around 0.5, so quite different. So there are a number of factors that come into play as we design our products with the new propellants. Looking at how these properties impact atomization and evaporation, and then how that in turns affect the APSD. APSD is a key metric. This is a chart from a interesting paper by Wang et al. From this year, looking at the atomization of droplets from MDIs using the 4 different propellants and with varying amounts of ethanol. And there's quite a bit of interesting stuff going on here. First thing you can see, as you increase ethanol in the formulation, the droplets, the size of the atomized droplets increase. Not a big surprise there. For the ethanol-free formulations, it's interesting. There's some data from Wang et al. The empty symbols represent some data Kindeva has generated. There are some differences, but the rank order is the same. The formulations with HFA 152A produced the largest droplets. HFO 1234 had a little bit lower, and then the current propellants had the lowest atomized droplet size. And it's a little counterintuitive in that HFA 152A has a higher vapor pressure than 1234 and HFA 227, but it produces larger droplets. And that just shows that there's other factors beyond just vapor pressure that impact the size of the atomized droplets. Another interesting thing here is that as you increase the ethanol concentration, the rank order of which produces the highest droplet size changes. So droplet size obviously impacts mouth and throat deposition, and there's some interesting differences in the propellants and different formulations. Okay, I've talked about droplet evaporation. Why is that important? Well, this is some work we published a number of years ago looking at a very broad range of MDI formulations. And we measured for those formulations the fine particle fractions from cascade impactor data. We also then, using evaporation equations, calculated how long it took for the propellant and/or the ethanol or whatever co-solvent you had to evaporate. And we saw a very strong correlation between the fine particle fraction and the time required for the droplets to evaporate. And that depended on the propellant properties as well as the properties of the co-solvent. If the formulation evaporated really quickly, you had efficient delivery, whereas formulations for which it took a while for the droplets to evaporate, the droplets are larger in that turbulent region in the mouth and throat, and you get more deposition. All right, how about these new propellants? In the paper by Wang et al., they did some calculations of the droplet lifetime for the 4 different propellants for formulations without ethanol. Combining that with some cascade impactor data on formulations without ethanol for these propellants, when you look at where those results, they fall pretty well in line with the trend we saw previously. And one thing to note is that there's a slight increase in the droplet lifetime for 1, 2, 3, 4, and that results in a little bit decrease in the fine particle fraction, and then a bit more for HFA 152A. So because of the evaporation properties of the propellants, we may see some decreases now in the fine particle fraction. If you add co-solvent then to those formulations, you move down on the curve and those differences in the propellants may go away. Okay, so let's look at some actual cascade impactor data so I'm going to show 4 different slides. We're going to look at beclomethasone solution formulations. The actuators and valves will be the same. We'll look at the different propellants, and the difference in the slides is really the ethanol concentration. And you can see that for formulations with 15% ethanol, there's really very little difference in the APSD from the different propellants. And the reason for that is that the performance is really being dictated by the ethanol and the time required for that to evaporate. And so you get really consistent fine particle fraction between the different propellants. All right, decreasing the ethanol concentration to 12%. You're starting to see some differences in the APSD profile, but still pretty similar. The fine particle fraction has gone up from 40% to over 50% because there's less ethanol there. If you decrease further to 8% ethanol, again, you're seeing a little bit more difference between the different propellants and a little bit more efficient delivery. Now, if you have no ethanol in the formulations, it's a very different profile. I'll explain that in a second. But the fine particle fractions, you have a pretty significant difference between the propellants in terms of the fine particle fraction. Now, the reason this profile is quite different is that in order to get solution formulations of beclomethasone in an ethanol-free formulation, I had to decrease the concentration of the BDP in the formulation from about 2 mg per mil to 0.05. So we have very small residual particles, yet you do see differences that for HFA 152, and you have the highest throat deposition and the lowest in the filter, followed by the 1, 2, 3, 4, and then the current propellants. So some differences in the performance when you change out the propellants, but it depends on the formulation. If you have a formulation with a lot of ethanol, you're not going to see that much of a difference when you change propellants, and you might be able to get away without having to do too much hardware optimization like actuator orifice diameter. However, for formulations without ethanol, you're likely to see increased some changes in the APSD, and you might need to do some fine tuning of the actuator, which that's all doable. These are differences. This is just the stuff we're going to have to work through. All right, changing gears from product performance. Let's look at a little bit at product design of the formulation, and in particular, the impact of solubility and how propellant can change that. Drug solubility is probably the first thing that MDI formulators look at when they get a new molecule that they're going to formulate. And you either want really high solubility, so you can have a solution formulation, or you want minimal solubility, so you can have a stable suspension. If you do have a solution formulation, drug degradation is one of the things you just need to address and evaluate and make sure it's not too much. For solution formulations, obviously you have to have sufficient solubility of the drug in the propellant formulation to achieve the dose you desire. But you really need to have, you can't formulate right at the solubility limit, otherwise you'll have, during temperature cycling, you'll see precipitation of the propellant. So you need to have enough solubility of the drug. So often ethanol is added to the formulation in order to achieve that. There are approaches like adding acid to address potential drug degradation issues. If you have a suspension formulation, solubility is kind of the enemy. You either want, you want minimal solubility because the partial drug solubility can lead to changes in the drug size due to either Oswald ripening or crystal transformation. Here are some measurements on a number of inhaled drugs for the different propellants. Some results, there are multiple references and both results are presented. But for the 6 different drugs evaluated, the highest solubility is for HFA 152A. That may be advantageous for solution formulations. For 1, 2, 3, 4, that tends to have very low drug solubility, and so that may be advantageous for suspension formulations. That said, you can, for example, for the 152 formulation of the first steroid, there's 0.01% weight drug solubility. If the drug you need in your formulation to get your dose is 0.5% by weight, having a couple of percent of drug in solution probably isn't a deal killer. You probably can get a viable formulation there. But if your drug concentration for your dose is 0.02% and half of your drug is in solution, that obviously wouldn't be a viable formulation. So drug solubilities do different in the propellants, and we need to evaluate that. Similarly, we do have some differences in excipient solubility in the propellants. In general, the excipients need to be in solution to do what we want them to do if they're a surfactant or a bulking agent. David Lechuga will talk about one exception with the aerospheres particles, but generally you're wanting the excipients to be soluble in the propellant. This was one of the challenges in the CFC to HFA transition. The excipients that were worked well in the CFCs didn't necessarily work well in the HFAs. In some cases, ethanol was added to solubilize those excipients. And we may need to do things like that again. So there are some difference in excipient solubility, and I'll show that in this next slide here. So here are some common excipients and their solubility in the various propellants. This is from visual solubility assessment. So if you have a number like less than 0.02%, that would mean we put in 0.02% by weight of that excipient in the propellant. You shake it for a period, and then you look, and if it's still visually there, the solubility is less than that value. In terms of numbers like this, where you have 1 to 2%, that would just indicate that 1% oleic acid was soluble in the HFA 152 propellant, whereas 2% was not soluble. In terms of trends on this chart, it's a little more complicated. So, for example, oleic acid is widely used. In 152, it has much higher solubility than in the current propellant. So that may be desirable. It may be a good excipient to use. However, tweens, 152A had much lower solubility of those in the propellant than existing propellants. And in general, HFO 1234 has fairly low solubility of the most excipients. So you may either need to decrease the amount of excipient you put into the formulation, or you may need to do things like add ethanol to the formulation. So these are some of the things that we need to work through as we formulate and design our MDIs with these new propellants. All right, what about the impact of propellant on the behavior of suspension formulations? Now, if you look at Stokes' law, the settling of particles in a fluid is determined by a number of parameters, including a term that is the comparative density of the particles versus the density of the formulation. And for most crystalline drugs that we've worked with, or I have in my experience, their density is typically about 1.2 to 1.3. That's a general rule, but others you can have more dense. But the density of the propellants, 227 has a higher density, so it will have a negative settling velocity. It will cream, whereas in 152A, for example, the density is much lower and the formulations will settle. So we often want to look at the formulations and see what they look like. These are pictures of a particular steroid formulated in the four propellants. No other excipients were added. So it's just a simple formulation. It's shaken, and then here's the appearance after 60 seconds. You can see the most of the drug has creamed to the top for the 227 formulation. For the 152 formulation, the drug has settled to the bottom of the vial, and then it's in the process of doing that for the 134A and 123 formulation. So visually, we like to look at these, but I can say from my experience that we've had formulations where they didn't visually look like good suspensions, but they give you very good reproducible dosing. So really it comes down to you need to measure the dosing. The appearance will look different, but I don't get too hung up on that. One other important thing for suspensions is that they can deposit on the surfaces of the canister or the valve. And the propensity to do that can be very different for the different propellants. Here is some data for formulations of albuterol sulfate and HFO 1234, and no other excipients were added. When we had no ethanol in the formulation, just drug and propellant, there was a significant amount of drug deposited on the surfaces of the can and valve, and that resulted in below target emitted dose. However, when a little bit of ethanol was added, that drug deposition went way down, and the delivered dose was much closer to target. So again, these are the types of formulation issues we need to work through as we transition. Okay, lastly, some of the impact of propellant properties on spray pattern. The spray patterns for the different propellants, they are different. Here is some data showing spray pattern for the different propellants at 3 and 6 centimeters. The blue symbols represent ethanol-free formulations. The red symbols contained 12% ethanol. And what you see is that for 134A and 152A, they have higher spray pattern area than the other two propellants. If you add ethanol, the differences between the propellants is a little bit smaller, but it's still there. So this has been shown in a number of publications, and it's due to the different properties of the propellants. Now, there are some things you can do to manipulate spray pattern. This is some GDUFA-funded research that looked at spray pattern for various formulations in HFA 227. And what it shows is that by changing the orifice length or the jet length, as it's sometimes called, you can reduce the spray pattern area. So there are some knobs that you can adjust, but they have an impact also on some of the other metrics like APSD, which are probably a little bit more significant. So these are the types of formulation and hardware optimization we're going to need to do. So in conclusion, there are going to be some differences in the pharmaceutical performance with these new propellants. APSD can be impacted by the propellant choice, but it does depend on the formulation. For example, the amount of ethanol in the formulation. And those are things that can be compensated for by changing your actuator design. There are solubilities, so the formulation approach is going to be impacted by solubility of both drug and excipient. So it won't always be so simple to just change the propellant. There may need to be other formulation changes that are needed as well. And then lastly, the different propellant properties do affect things like the suspension, settling behavior, and spray pattern. So thank you very much for your attention, and those are some of the things we need to deal with.
Okay, our next speaker is from FDA. It's Dr. Stacy Chen. She's an allergist and a clinical team leader in the Division of Pulmonology, Allergy, and Critical Care. And she has been working on the low global warming potential propellant transition programs. She's going to be providing an introduction to the O&D framework for the transition. So please welcome Stacy Chen. Thank you, Stacy.
All right, good morning. My name is Stacy Chen. As Sally mentioned, I'm a clinical team lead in the Division of Pulmonology, Allergy, and Critical Care in the Office of New Drugs, or O&D, at FDA. So today, I will be providing an introduction to the O&D framework for the LGWP propellant transition. Here are my disclaimers. I have no conflicts to disclose. So to provide some context for how O&D is approaching this propellant transition to the branded drugs, I'd like to highlight the following points. LGWP propellants are new excipients for inhalation, and there are no FDA-approved NDIs containing either of the LGWP propellants in development. Because MDIs are locally acting, there is some uncertainty in how the new propellants will affect drug deposition in the lungs and whether this may impact safety or efficacy of the product. That said, as you've heard, based on available data, the chemical properties of the approved HFA propellants appear to be relatively similar to the LGWP propellants, more so than the CFC and HFA propellants were in the previous transition. However, it is still too early to tell whether there will be any novel issues with the new LGWP propellants. Now, I will review some of the key concepts that inform our framework for LGWP propellant product development. So MDIs containing LGWP propellants are considered new products. Historically, new inhalation products have required full clinical development programs to support a marketing application. But given what we currently know about the LGWP propellants and their similarity to approved HFA propellants, as well as the recent experience with generic MDI development, we do believe there is an opportunity here to streamline clinical development with this propellant transition and to hopefully minimize the impact on supply chains, the market, and patients. As we've been formulating our framework, we've identified two main scenarios. One, there are approved MDIs that switch out the existing HFA propellant and make any other necessary changes to the formulation or device. in order to incorporate the new LGWP propellant. In this case, the primary intent is to replace the currently marketed product with the new LGWP propellant MDI, and the products would essentially have the same dose, the same name, indication, labeling, etc., with only minimal changes to the user interface and device. In this case, an abbreviated clinical program is likely feasible. The second scenario consists of a previously unapproved MDI that is being developed for the first time with an LGWP propellant. As I mentioned, new inhalation products typically require full clinical development programs that include dose ranging, efficacy and safety trials for each indication. However, there may be some products in this scenario for which a clinical program might be possible if the sponsor can demonstrate sufficient comparability between the proposed product and the listed drug in order to rely on the agency's previous findings of safety and effectiveness. However, this determination will be made on a case-by-case basis. In the remainder of my presentation, I will provide a high-level overview of our current thinking about the streamlined approach to the development of LGWP propellant MDIs, and specific details will be discussed in the presentations that follow in this session. Because we are still early in the transition with no approved LGWP propellant MDIs, our advice may and likely will evolve over time. The data needed to support a streamlined approach should be generated from comparative studies of the approved HFA reference product and the proposed LGWP propellant test product. So when you hear about the studies we're looking for, it's these comparative studies of the two. We really strongly recommend taking a stepwise approach to development, and I'll walk through these steps in the next few slides. We also encourage early and frequent interaction with the agency because each program has unique considerations, and as we're still early in this transition process, we expect our thinking to evolve as we gain more experience and more approvals and work through different issues. So the LGWP propellants are subject to the standard non-clinical data study requirements. However, we anticipate that this data will be obtained by the propellant manufacturers and included in a drug master file or DMF. So individual drug sponsors may acquire a letter of authorization to obtain right of reference to the drug master file or DMF so that the agency may then access and rely on information contained within it. Therefore, the first major step of development for an individual product is to generate product quality data. We expect to see a robust comparison of the critical quality attributes between the approved HFA MDI and the proposed LGWP propellant MDI. This should include an assessment for bioequivalence. And we understand that these comparative analyses are not typically conducted for new drugs, but demonstrating in vitro similarity is key for pursuing a streamlined development program. And to be clear, we are not requiring in vitro bioequivalence, but think it is important that we have a thorough understanding of the similarities and differences between the products. And as you've heard in the previous presentations, we expect that there will be some differences because just the different chemical properties of their propellants. However, we expect some justification for these differences and whether or not they're anticipated to meaningfully impact safety or efficacy of the product. That said, if there are major differences observed, a streamlined program is likely not appropriate. Provided that the in vitro data are comparable, the next step would be to conduct PK relative bioavailability studies. And depending on the API, PK studies might need to be conducted with and without charcoal block. More details will be covered in the clinical pharmacology presentation that follows. Similar to the product quality assessment, there should be an assessment for bioequivalence between the approved HFA reference product and the proposed LGWP propellant test product in the PK studies. Again, this is not routinely performed for new drugs, but demonstrating sufficient comparability is key for supporting a streamlined clinical program. Although bioequivalence is not a requirement for new drugs, it is the goal that sponsors should be aiming for. The last step in development consists of clinical studies. Provided there's adequate in vitro and in vivo evidence demonstrating sufficient similarity between the approved HFA MDI and the LGWP propellant MDI, the clinical program can be streamlined. At this early point in the transition, the minimum requirements include a comparative clinical endpoint study, such as a pharmacodynamic bronchoprovocation study or a short lung function study, as well as a long-term safety study to support the safety of the new propellant. The limited scope of the clinical program is contingent upon showing similarity in the previous steps. So I've outlined the scenario in which a streamlined program would be acceptable. However, there are situations in which a full clinical program might be needed, such as when the in vitro product quality data or PK data are not comparable between the approved HFA MDI product and the test LGWP propellant product. If there are major changes to the formulation, the dose, device, or user interface between the products. And finally, as I mentioned, unapproved MDIs that are being initially developed with LGWP propellants generally have needed full clinical development programs. So this includes dose ranging, efficacy safety trials, unless sponsors are able to demonstrate comparability to the listed drug by conducting a BE assessment of the product quality and PK data. We think that this situation might be more common early in the transition, while clinical studies are still needed to support efficacy and safety of the change in propellant. Applications that might otherwise have been abbreviated NDAs would need to be submitted as original NDAs because of these clinical study requirements. So to summarize, FDA's approach to the LGWP propellant switch for brand drugs. Streamlining the clinical development program, primarily by reducing the number of clinical study requirements, is a possibility. However, the acceptability of this approach depends heavily on demonstrating comparability in the product quality and PK characteristics between products. Although bioequivalence is not a requirement for new drugs, sponsors should consider BE the target to aim for when proposing a streamlined approach. We are still early in the transition and expect that our approach and advice will evolve as we gain more experience and approvals. There are some scenarios in which a full clinical development program may be needed. And I recognize that the regulatory submission pathway is an important topic of interest to this audience, which I have not covered in this talk. But because of this, we encourage early and frequent interaction with FDA to discuss your individual programs. We believe we're best able to provide advice on scientific and regulatory issues within the context of individual applications. So thank you for your time and attention. I've provided the general overview and framework from the O&D perspective, and you'll hear the details in the presentations that follow.
Thank you, Stacy. Okay, next we're going to hear from Dr. Luqi Pei, who's a master pharmacologist in the Division of Pharmacology Toxicology that supports our review division, and he has an expertise in inhalation products. So thank you, Luqi.
Again, my name is Luqi. I'm a non-clinical reviewer supporting the pulmonary and critical care division. And I'm going to talk about the non-clinical considerations for the transition of low global warming potential propellants. Here's my disclaimer. I don't have any conflict of interest to claim. I'll be covering basically 3 topics. One is the non-clinical requirements and guidelines for the development of excipients, and then the principles we use to evaluate those propellants. And also finally, with the lessons we learned from the last transition period. There are regulatory requirements for non-clinical development of excipients. Here's the propellants. The requirements are listed here. That's in the CFR, part 312 and 314, and also the GFR and the good regulatory, good practical, good, laboratory practice guidelines. But the more information is covered in the FDA's guidelines. There are many guidelines available on the website, but I here just listed 2 overarching guidelines. One is the excipient guidance published in 2005, and then the RCHM 3.2, that's overarching guidelines for developing of drug products. There are many other guidelines available, but I'm not going to discuss it in detail. They are on the website you can refer to. And the general principle of non-clinical evaluation is that those propellants are expected to be widely used once approved. They will be used by millions of people for decades to come. So we're expecting a complete, thorough non-clinical characterization of the toxicity profile of those excipients. Currently, there are 4 propellants in interest. 2 of them are already on the market. That's 134A and 227. And then 2 are in the development. That's 152A and 1234A. Though the development of these propellants have been in discussion for at least a decade, we have been discussing actively with some manufacturers and drug sponsors. But these discussions are happening in 2 areas. One is the drug product development, the sponsor. Those discussions are happening in the IND and NDA applications. Those discussions are generally product specific, because different products may have different clinical use, different population, and different risk benefit. Then the major development is actually discussing under the drug master file. That's the manufacturers submitted the non-clinical data to the drug master file, and we'll review it over there. And then the IND and the NDA sponsors obtain a right of reference for us to look into those data. Because those non-clinical data are proprietary information, and I cannot discuss it here, but here's the general information that what the non-clinical program look like. It'll need to cover cover the pharmacology basically for propellant. It's a lack of pharmacological activity. We need to look at the pharmacokinetic profile of the compound, and then the major part of it is the toxicology. Toxicology, you need to cover the general toxicology, the reproductive and development toxicology, the genetic toxicology, and the carcinogenicity. There are other data applicable too, but I'm not going to go into each specific area. Because we have been in the contact discussion with the developers and sponsors, and some of the considerable progression has been made, and some of the information are available, like the publication by Giffen et al. In this year's SOT, and it's also published in the, I think it's International Journal of Toxicology, and you can reference to there. And of course, there are the discussions are still ongoing. The progress may not be the same, some are more advanced than others. So last one is the lessons we learned from last round of switch. We learned from last round of switches that the HFA propellant increased the bioavailability of active pharmaceutical ingredients, generally by a third. And another thing we learned is that although the increased bioavailability, but it does not change the toxicological profile of those drugs. So with the previous lessons we learned, we think that in this round of switching, we can reduce the non-clinical requirements for the transition. Example is that for the previous round, we asked for drug-drug interactions toxicology studies in the non-clinical program. And this round, we think that those studies may be waived, we do not require that. And that's pretty much my talk. Thank you.
Okay, thank you, Luqi. It's my pleasure to introduce the next speaker, Dr. Craig Bertha. If any of you have developed inhalation products, Craig has probably reviewed your application. And he's been at the FDA for decades. He's a CMC reviewer. He's got incredible subject matter expertise in this area, and we're very fortunate to have you on board for this next transition. So he's going to be discussing quality considerations for the low global warming potential propellant transitions.
So, good morning. Let me go back here. All right, so Sally said I'm going to talk about the quality considerations for transitioning to the low global warming propellants for brand products. And this is my disclaimer, and I also don't have any conflicts of interest to disclose. So some of what I might talk about might be redundant, but bear with me. I'm only going to be up here for about 10 minutes. So propellants provide the bulk of the MDI's formulation, and they either dissolve or disperse the drug in the formulation. And as we've heard before, especially from Stephen's talk, the physical chemical properties are going to be the major determinant of the properties of the delivered aerosol. The propellants rapidly expand and evaporate as the liquefied propellant is released from the valve and through the actuator, and they propel the formulation through to the patient and ideally produce droplets in particle size that are in the range suitable for inhalation into the lungs. This slide just shows the critical quality attributes in bold that are related to the product performance in terms of dose delivery and APSD. So the shot weight, the delivered dose uniformity, aerodynamic particle size distribution by cascade impaction, and spray pattern testing. And Markham had this slide earlier. I just wanted to reiterate some points that maybe had already been made. In the first generation propellants, the differences between the physical chemical properties between those and the second generation propellants were quite distinct. And luckily this time, the differences between the second and the third generation propellants in terms of physical chemical properties are not as drastically different. And at the bottom there in green, you can see the two propellants we've been talking about today that are the two current candidates for low global warming potential. On this slide, in the first bullet, I've just shown the global warming potential for the old propellants, the second generation ones, and the new, and you can see that the global warming potential is a lot lower for these new propellants, particularly for HFO 1234ZE. And in the second bullet, where I have the physical chemical properties listed, you can see that HFA 134A and 152A are quite similar. They have similar boiling points, similar dipole moments, which is an indication of their polarity, and similar solubility of water in those propellants. Whereas HFC 227EA and 1234ZE have a similarity in terms of their boiling point, their dipole moment, and the solubility of water in them. There are some distinct differences, as Stephen had noted in his talk. For example, if you're switching from 227EA to 152A, there's a drastic difference in density between those two propellants. But one good thing is that the vapor pressure of the two new propellants, HFA 152A and 1234ZE, lie between the vapor pressures of the second generation propellants. So basically, the bottom line is that the differences, as we've heard before, between the physical chemical properties of the new third generation propellants aren't as drastically different compared to the second generation, which might make the transition easier than the first transition from CFCs. But still, as Stephen had noted before, there could be drug solubility differences, so you may have to switch from a suspension to a solution formulation or vice versa. You may need to use different amounts or co-solvents or different amounts of co-solvents. You may need different surfactants or other suspension aids. And as Stephen also mentioned, internal canister coatings may be needed if suspended drug particles are sticking to the inside of the canister or the valve components. Then the density differences are also going to affect the creaming or the settling of the suspensions. Valve components may need to change. If there's a high vapor pressure, you may need more robust gaskets to lower the leak rate or prevent moisture ingress. Obviously, leachables will have to be characterized and controlled because if you're changing plastic and elastomeric components, you're probably going to have a different leachable profile, obviously. If you're making changes to the valve for whatever reason and it changes the force of actuation, then you may have to tweak your counters if change the force to count so that you don't have undercounting or significant overcounting of the counter. But at the end of the day, it's basically going to depend on the formulation you've chosen and the propellant that will determine how the dosing performance, aerodynamic particle size distribution, and the spray pattern, plume geometry look compared to the previous products that you're switching the propellants with. And then, as Stephen also mentioned and Sally mentioned, there could be different cleaning requirements with these new propellants and priming requirements that may need to be incorporated into the labeling for the patients. HFA 152 is said to be somewhat flammable, so that might mean that manufacturing changes at the plants will need to be different, which could impact patients' disposal procedures, and that might be something that might need to be captured in labeling as well. And as Sally mentioned, there could be different patient perceptions on how it feels when they use the new product relative to the ones they're used to using with the second generation propellants. But at the end of the day, from a CMC quality perspective, as Sally had mentioned, an MDI with a new low global warming potential propellant is essentially a new MDI product. And from a biopharmaceutics perspective, as Stacy had mentioned in her slides, we're recommending that companies do the in vitro comparisons that are outlined in our product-specific guidances that OGD uses as a part of their determination of bioequivalence for generic products. And the first bullet here just lists some of those in vitro studies. So we recommend that you consult the most appropriate PSG for your product and then evaluate it in terms of that bioequivalence approach. And hopefully the results will be comparable so that maybe you can reduce the scope of your clinical program. And if there are any differences, they would need to be justified, as to why they might not signal that there would be different in vivo deposition compared to the product with the second generation propellant. So the submissions for an MDI product that would come in whatever form that's going to be, either NDA or a supplement or whatever, really should include all the data for CMC that we need. But of course, if you've already submitted some of that data, you don't need to resubmit it. You can just refer to your previous submissions. In case you use the same drug substance, we don't need to re-review that. No need to reinvent the wheel. So now I'm just going to go through the different sections of the NDA, and what I have highlighted in blue are just some of the things that where you may need to supply additional information. So for example, for the drug substance, if you're going from a solution to a suspension MDI, then you may need to, well, you will need to characterize crystalline form. You'll probably have a micronization process that you'll have to describe and control. There might be a formation of amorphous drug substance because of the micronization process. Perhaps you'll need conditioning, so that would all need to be, explained in the applications. And there would need to be a characterization of particle size distribution, and the stability of the micronized drug would need to be studied as well, because it might impact the stability when you make such small particles. And there could be other things too. If your drug substance has a secondary amine, maybe the micronization and the nitrous oxides in the air would form some NDSRIs, or, the mill could increase the levels of elemental impurities, things like that. So that's one area where you may need to submit more information than what you would have already submitted in the product with the previous propellant. Of course, the formulation would need to be described, and the pharmaceutical development section probably will have to outline all the development aspects that led to the final chosen configuration. And then we would like to see the characterization studies for these MDI products again. That's something that would need to be there. And obviously the batch formulation and the manufacturing processes are going to be new. As far as the propellant, Luqi mentioned that there probably will be type 4 DMFs for these. So in those DMFs, from a CMC perspective, we would need, characterization studies and specifications, batch analyses for those, and, information about impurities, because we work closely with our pharm tox colleagues to make sure that the levels of those impurities are safe and acceptable. And then there would need to be analytical tests that were validated in that DMF as well for release of that propellant, justification of specs and stability data. But these are inert propellants likely, so stability probably won't be an issue. In terms of the drug product, of course, pretty much need everything there. You need specs, probably mostly new analytical methods, but you probably can use similar analytical methods that you used for the previous product. So that might be where you can save some time. Validation data, obviously, batch analyses and justification for specs. Now, if you're not changing the container closure system, then you could just refer to the previous DMFs that you had referred to for your old application. But if there are changes, then we would need to review those. And of course, we would need stability data for the new product. It could have, if you're going to a solution from a suspension, it may be more chemical degradation. If you're going from a suspension to a solution, no, if you're going to a solution, there may be more chemical degradation. If you're going to a suspension, there may be physical chemical problems like creaming or settling, things like that. So that would be one of the aspects of it. Plus, leachables as well would be a part of your stability program. So in summary, once you switch out the propellant, you basically have a new product, and you're going to need to submit probably most of the CMC information that you normally would for a new MDI. But of course, you can reference previously submitted information if it's applicable. So I'd like to thank you for your attention.
Thank you, Craig. Our next presentation, I'm delighted to call up Dr. Sneha Dhapare, She is a senior clinical pharmacologist in the division of clinical pharmacology that supports our division, and she's going to talk about the clinical pharmacology considerations for the transition.
All right. Good morning, everyone. As Sally mentioned, my name is Sneha Dhapare. I'm a senior pharmacologist in the Office of Clinical Pharmacology at the FDA. And today I'm going to talk about clinical pharmacology considerations for low global warming potential propellant transition for metered dose inhalers. Okay. This is my disclaimer for this presentation. And here's the outline. So I'll start by talking about the previous CFC to HFA propellant transition and then lessons learned from a clin pharm perspective. And then we'll go into details of the clinical pharmacology considerations to establish bridge between the HFA and the LGWP propellant-based MDIs. So we'll talk about pharmacokinetic or PK considerations as well as pharmacodynamic or PD considerations, particularly for the short-acting bronchodilators. So let's talk about a phase out of CFCs. As Sally noted in her talk, the phase out of CFCs provide a good background for us to understand how the change in propellant, in that case from CFC to HFA, affects the MDI development. So CFCs were phased out following the 1987 Montreal Protocol, and MDIs were reformulated considerably due to the differences in chemical properties between CFCs and HFAs, as we have heard before. So this included changes in strength, changes in excipient, and changes in delivery device, all of which may affect the drug systemic exposure. So generally, transition from CFC to HFA MDIs included these elements in the clinical development program. They included 2 phase 3 efficacy and safety studies with HFA MDI in patients that may be asthma or COPD patients, long-term safety studies in patients, comparative efficacy and safety studies between HFA and CFC MDIs in patients, relative bioavailability study to assess systemic exposure of HFA compared to CFC in healthy participants and in adults. Moving on to the current HFA phase down, as has been noted before, this is a phase down that was implemented following the Kigali Amendment to the Montreal Protocol in 2016. MDIs are currently exempt. Alternative low global warming potential propellants have been investigated but are yet to be approved in an FDA inhalation product. Several alternatives have been studied, like HFA 152A and HFO 1234ZE, and they appear to have more comparable physicochemical properties to the current HFA propellants, like the HFA 134A, as was talked about by Dr. Stein. Research suggests that LGWP propellant MDIs with comparable properties to their HFA counterparts may be achieved by selection of an appropriate LGWP propellant, changes in device, so for example, valve or orifice diameter, as well as changes in formulation, like addition of a co-solvent. So then let's move on to talking about pharmacokinetic considerations for establishing a bridge. First, I'll talk about general PK concepts for MDI products. The role of propellant in MDIs is complex. It's more than just an excipient. It's a solubilizing medium for formulation. It's a driving force for the release of product from the actuator. It atomizes the formulation into droplets, and it results in droplet drying and resulting deposition in the lung. So these properties affect product performance, and therefore it affects regional lung deposition. PK assessments for an inhalation product are usually downstream of the lungs, so that's the site of action. Therefore, traditionally, PK studies inform systemic safety of inhalation product. This is a very simplified and general overview of what happens to the dose that is released from an MDI actuator. So typically, we see a lot of oropharyngeal deposition, and a major part of which is swallowed and goes into the GI tract. This may be in the range of 70%, depending on formulation and patient factors. Some part will go to the trachea, bronchi, bronchioles, and alveoli in the lungs. Eventually, from the GI tract and from the lung, the drug will be absorbed and will end up in systemic circulation. The drug can be eliminated by local elimination, exhaled, or mucociliary cleared. Therefore, what we usually measure in a PK study is the lung levels in systemic circulation, and they're often confounded by this portion that comes from the GI tract, and not just the lung. So one way to solve the piece of this puzzle is to block the GI absorption by the use of charcoal. So what are charcoal block PK studies? Oral intake of activated charcoal in PK studies is aimed to prevent the gastrointestinal tract absorption of the active ingredients. The resulting systemic PK data may be considered due to absorption through airway exclusively. This separation of non-airway absorption from airway absorption may help interpret the systemic exposure, particularly for assessing product similarity. Here are some key considerations for a charcoal block PK study. The study design is usually similar to a relative bioavailability study without charcoal. A higher dose than relative bioavailability study may be needed if substantial portion is swallowed or absorbed. Absorption efficiency of activated charcoal is dependent on the physicochemical properties of charcoal. Therefore, the charcoal dose should be adequately justified. This is applicable to MDIs containing drugs with substantial GI absorption. So for example, the albuterols. This may not be applicable to MDIs where there is negligible GI absorption, for example, epinephrine. Therefore, from a clinical pharmacology perspective, relative bioavailability studies comparing exposures between new and the old propellants can support assessment of product comparability. In some cases, PK studies with charcoal block may provide additional supportive evidence for comparability in the lung delivery. Here's an example of the PK study design. It is a single-dose crossover study with appropriate washout. Treatments may include MDI with HFA and MDI with LGWP propellant. Subjects are healthy adults, and the dose is minimum number of inhalations required for the detection of systemic exposure using a sensitive bioanalytical assay. The study should be sufficiently powered for establishing BE, and BE analysis should be performed to provide conclusions on PK similarity. Relative bioavailability studies should be conducted with the to-be-marketed product. Study designs can vary with different MDI products, and therefore early communication with the FDA is important. Then let's move on to the pharmacodynamic or PD studies. Lung function-based PD studies provide confirmatory evidence to support similarity. An example of a PD study is a properly designed bronchoprovocation study for albuterol that has been used in ANDAs to support approval. This bronchoprovocation study includes administration of differing doses of albuterol on separate days in a crossover study design with subsequent challenge with methacholine to provide a bronchoprotection dose-response curve. Depending on the drug class, complexity of the product, as well as the development program, the type and PD study design may vary. Here's an example of the bronchoprovocation study. This is taken from the albuterol PSD. A study design is generally single-dose, double-blind, double-dummy, randomized crossover study with appropriate washout. There can be 4 potential treatment periods. I'm not going to go over this in detail because you can find this study in the PSG that's referenced below. So to summarize, from a clinical pharmacology perspective, PK comparison studies provide supportive evidence to demonstrate comparability between products. The scope of PD studies depends on the drug class, the complexity of the product, and the drug development program. Early discussion with FDA is important, and the clinical pharmacology approach may evolve as we gain more experience with the approval of LGWP propellant-containing MDIs. I'd like to end my presentation by thanking my wonderful colleagues from FDA, some of whom you have heard from here today. Special thanks to Dr. Jan Zauren, who is the team lead in the pulmonary product, for his expertise and guidance. Thank you so much.
Thank you. Okay, our last presentation, and we are right on time, is from Dr. Aishah Ali. She's an allergist in our review division. She's a senior physician, and she's going to talk about clinical considerations for the transition.
Thanks, Sally. Do I just to go forward? Okay, perfect. All right. Good morning, and thank you for all being here today. My name is Aishah, and I'm a senior physician in the Office of New Drugs, Division of Pulmonology, Allergy, and Critical Care at the FDA. Today, I will be discussing the clinical considerations for the LGWP transition of existing brand name products from the perspective of the Office of New Drugs. I have no financial conflicts to disclose. We will begin with some general considerations to keep in mind as we discuss the clinical approach to the propellant switch programs. As mentioned, no MDIs with LGWP propellants are approved at this time. We are presenting our current thinking, noting that we are in early stages and there may be an evolution in thought as we gain experience through the review of more programs. We anticipate that clinical data requirements will change once we have approved LGWP propellant MDIs and more safety data is reviewed. As each propellant switch program will be unique, we encourage early and frequent interactions with the FDA for alignment. We anticipate the LGWP propellants to be more similar to current HFAs as compared to the previous CSC transition as mentioned. Sponsors have been proposing to transition from approved HFA MDIs to new LGWP MDIs using the same trade name and label. Streamlined clinical programs are designed to evaluate for differences between products. This approach is different than a typical new MDI development program, and we will also be considering experience and knowledge gained from generic MDI development programs to help guide our approach when appropriate. Over the past couple of decades, our OGD colleagues have worked on development programs for generic MDIs and have invested in research for generic MDI drug development. They have issued several FDA product-specific guidances that outline the agency's current thinking and expectations on how to develop generic MDIs that are therapeutically equivalent to listed MDI drug products. These FDA PSGs will help provide the framework for LGWP MDI products that are proposed to be therapeutically equivalent to an approved HFA MDI. As discussed, a stepwise approach for the propellant switch program should be utilized. As you have heard, the foundation of the program will be a comparison of the critical product quality in vitro data between the LGWP MDI and the approved HFA MDI. This comparison should include an assessment for bioequivalence. We anticipate that there could be in vitro differences between the LGWP MDI and the approved HFA MDI. If differences in the in vitro data are noted, sponsors will need to justify or provide support that the differences do not impact efficacy or safety of the new product. The next step is a comparison of the PK data between the LGWP MDI and the approved HFA MDI. This comparison should include an assessment for bioequivalence. If differences in the PK data are noted, sponsors will need to justify or provide support that the differences do not impact the efficacy or safety of the new product. The device and user interface of the new product should also have no significant differences. A streamlined clinical program will be contingent upon the similarity between the in vitro and PK data. If you are noting significant differences between the products, you should discuss this with the agency, as a streamlined program may not be appropriate. The main components of a streamlined clinical program include a comparative efficacy study, a long-term safety study, and a comparative use-related risk analysis. The study design of the comparative clinical study will depend on the active pharmaceutical ingredients in each product. The treatment arms for the efficacy study should include the LGWP MDI as a test product, the approved HFA MDI as the reference product, and an assay sensitivity arm, for example, placebo. Lung function efficacy endpoints are acceptable for the comparative clinical endpoint, and exacerbation data is not required for efficacy assessment. Local tolerance studies with propellant only, for example, ciliary function or airway sensitivity reaction studies that may be requested by other regulatory authorities, are not required. Long-term clinical safety data are required at this time, and the focus should be on demonstrating the safety of the LGWP propellant, as the safety of the active pharmaceutical ingredients was established in the HFA MDI program. When considering safety study, The design and duration depends on the active pharmaceutical ingredients. For example, the study design of a product intended as a reliever medication will be different than the product intended as a maintenance drug. We recommend obtaining controlled data with the LGWP propellant MDI, including the active pharmaceutical ingredient. However, propellant-only designs may be considered in certain cases, which will be later noted in this presentation. We anticipate clinical data requirements will change once we have approved LGWP MDIs and sufficient safety data for the novel propellants. In terms of the indication and population to be enrolled in the clinical trials, if an existing product has approved for multiple indications, the clinical study may be conducted in the most sensitive population. Pediatric patients may be included but are not required in clinical studies. The same age indications as the reference product may be applied if comparability is demonstrated. If no meaningful differences are demonstrated, all indications and labeling claims could carry over. The Pediatric Research Equity Act, or PREA, is triggered by the following changes to an existing product: a new indication, a new dosage form, a new dosing regimen, a new route of administration, or a new active ingredient. We do not anticipate that PREA will apply to the propellant change programs for existing approved MDIs, but will apply to novel, not yet approved MDI products using the LGWP propellant. We do note that each program is unique, and whether PREA is triggered is something we will determine on an individual application basis. Lastly, the to-be marketed product should be used throughout the development program, for example, to evaluate in vitro, PK, and clinical data. Now we will present two examples of study designs for the LGWP propellant switch. We will present a program for a reliever product followed by a program for a maintenance product. We encourage that all potential study designs are discussed with the agency for alignment. We will start with an example of an albuterol MDI. For the comparative clinical endpoint study, a pharmacodynamic bronchoprovocation study should be completed. An acceptable design would include a single dose, double-blind, double-dummy, randomized crossover design in an asthma population. For endpoints, post-dose PC20 or PD20 endpoints would be clinically relevant. To assess for bioequivalence, we recommend a dose scale analysis of pharmacodynamic data. For the safety study, a randomized, double-blind, active control design of 12 weeks duration could appropriately assess safety. For the treatment arms, we recommend albuterol with the LGWP propellant MDI compared to the approved albuterol MDI, but we may accept a propellant-only MDI study to demonstrate safety. For dosing, we recommend scheduled administration, for example, 2 inhalations 4 times a day and not only as needed, to ensure adequate exposure at the maximum recommended dosage. We refer you to the albuterol PSG for further study design considerations, and that's linked at the bottom of this slide. Next, we will present an ICS-LABA MDI example study design. Here, you can consider a randomized, double-blind, placebo-controlled, multi-dose design that is parallel group or crossover. The duration may depend on the design, but at a minimum, we expect 4-week treatment periods. The treatment arms here are the LGWP propellant MDI, the approved HFA MDI, and placebo for assay sensitivity. The sensitive population could be asthma. For endpoints, the primary endpoint could include peak FEV1 or serial FEV1 time curves, so AUC 0 to 12 or 0 to 24 hours on day 1, and trough FEV1 at the end of the treatment to assess the contribution of each component. For assessment of bioequivalence, you should determine the 90% confidence interval for the test to reference product ratio for the primary endpoints. For a safety study for ICS-LABA, an acceptable design would include a randomized, double-blind, active control study with a minimum duration of 24 weeks. Of note, this is our current thinking, and this may evolve as more programs are reviewed. The treatment arms in the safety study should include the LGWP propellant MDI as one arm, the approved HFA MDI for comparison. We refer you to the following product-specific guidances at the bottom of the slide on ICS LAVAs. The last topic for today is human factor studies. For streamlined programs, we expect no differences in the device and in the user interface between the LGWP propellant MDI and the approved HFA MDI. If the changes from the reference product are substantial enough to impact the user interface, the clinical program may need to be more extensive, as use-related risk analysis should evaluate for errors and risks related to the device. Comparative analysis should include labeling comparisons, comparative task analysis, and a physical comparison. A human factors study may or may not be necessary, and that determination should be made depending on the differences between the two products. If human factor studies are needed, a streamlined clinical program may not be appropriate. In conclusion, what we have reviewed today is our current thinking on streamlined clinical program requirements at this early phase of the transition of LGWP propellants. The stepwise approach should include an assessment of bioequivalence at each stage, as this is essential to determining acceptability of a streamlined approach to clinical development. We anticipate that the clinical data requirements will change once we have approved LGWP MDIs and sufficient safety data has been generated. However, each program is unique in its details, and our guidance may evolve with time. As reinforced throughout our talks today, we encourage early and frequent communication with the FDA to discuss study design components and reach alignment with the agency. I thank you for your attention today, and we welcome any questions from today's FDA presentations at the panel discussion.
Thank you, Aishah. We're going to get set up for the panel discussion here. I'd like to call back Luke from the EPA, Stein from Condiva, and also Hailin Zhang, who I mentioned earlier is going to be joining us as a panel member. She's a director of the Division of Pharmacology, Product Quality Assessment 12, and Biopharmaceutics. So she's going to provide the biopharmaceutical perspective. And then Banu Karimi Shah, she's going to be you're going to come up to okay, and help us with some of the online questions. I think if you have questions in the room, there's a mic going around. There's also a microphone here. Rick, why am I not surprised? You're first in line.
I'm trying to find information on whether mixtures of propellants with or without alcohol follow ideal behavior according to Henry or Ralph's law.
I'm trying to think of data on the new propellants, whether there's some data in the public domain, and I'm not sure if there's published data on that. So maybe somebody else can point us there.
Thank you. Next.
Yeah. Hi, this is Lloyd Brito with the Healthy Airways. Dr. Markham Luke started off the morning today asking about what is it that is giving the industry a lot of pain and giving us a lot of headaches. So what you're seeing being published over the last 3 or 4 years is that companies are trying to get bioequivalence in vitro established, and it's been a little bit of a struggle. So when delivered dose or particle size distribution performance is not acceptable, they try to modulate that by changing the actuator, by changing the formulation composition, etc. But when they're trying to do that, the design space around these variables continues to shrink and shrink and shrink. So is there a possibility that if you try to stack up what are the things that are most desirable and you say particle size distribution, deliver dose, and then say that because you're changing the propellant, there's going to be a change in the spray pattern, and plume geometry. So in that case, do you need to have in vitro bioequivalence established for those two parameters? But is it okay to say that this is the space that we are right now with those parameters, with the current propellants? With the new propellants, the reference point could change.
So I think this is a question maybe for Craig and Helene maybe to provide some input on. And I will say that there may be some case-by-case issues, and that if you're experiencing challenges with your development, this might be something you want to come and discuss with us to provide specific advice for your program. But Helene, I don't know, or Craig, if you want to.
Yeah, sure. So I think it's it's important. We do expect some differences for some of the physical properties you just described. In order to really compare the new formulated product with your existing product, it's important to compare them and have a full understanding. We also think it's important for the sponsors to understand the correlation between some of the in vitro properties to their in vivo performances. I think in one of the correct slides, when we mentioned we expect some differences. However, we also expect our sponsor to understand the differences and how they could impact. If their in vitro, in vivo correlation can be established, then it will depend, of course, on the case-by-case basis to see if the certain difference will be allowed, for example, for plume geometry or spray pattern. So we need that data to have that kind of understanding in order to really make that determination.
Okay, thanks. So basically what you're saying is if there are differences, then you have to do some in vivo studies to establish that those differences are not really impacting the clinical endpoints in any way. So.
I think we're going to take a very holistic approach. Of course, maybe abbreviated in vivo data sometimes are required. We also look at, for example, other in vitro approaches, even like in silico model approaches. But whatever, we look at in the submission, all the approaches or methods, you try to establish that in vitro and in vivo correlation has to be fully validated and has to be sensitive enough to build that kind of safe space.
Thank you.
Hi, thank you for your presentation. And my question is related to specification setting. In case of a streamlined program with a limited number of clinical batches, would you consider the opportunity to use batches coming from data of batches coming from release and stability, not using clinical trials, but representative of the to be marketed product?
All right, I think that from the CMC quality perspective, if it's a topically active product in the lungs, then we want to have a link between the data from the clinical batches compared to what you're going to be marketing. So you would still need data for the clinical batches in your application. Release data to compare to the registration batches.
Okay, so we can put all together if they are comparable, clinical batches and registration batches.
Sometimes even some of the registration batches are used in the clinical studies. That's fine.
Okay, thank you very much.
Yeah, I think that would be the preference. That would be the preference.
That would be preferable.
I would like to be sure I got the point. So basically, could I expand my data source, adding registration batches that were not using clinical, but are comparable to the clinical batches?
Well, I think you need to, we need to have that link from the clinical batches to the ones you're going to be marketing, and the representative ones are the registration batches. So if you're not going to have any clinical batches, in your, you need to have the batch analyses data,
yes,
but
in
you're
addition.
talking about stability data, right? Yeah. I would think that if you're using the same formulation and the same container closure systems, I don't think you necessarily would need, but it would be helpful if one of the registration batches was also a clinical batch. Okay. Let me put it that way.
Thank you very much. Very
Okay.
kind.
Thank you for a nice talk. So my question is to the FDA panelists. So you have summarized all the quality guidance on CMC clinical and non-clinical perspective for the LGWP transition. Is FDA planning to launch any official guidance on this consideration soon?
I'll take that. I think at this point, we are so early in the transition, and there's so much potential for evolution and thinking, and our expectations for particularly the safety data, I think we anticipate will change. So at this time, we don't have plans for a guidance. I think there's just too much in motion right now, and it takes a little bit of time to get the guidance out. I would worry it would almost be outdated by the time we get it out. But I just encourage you, as we've said over and over again, to keep interacting with us on your program, and we'll provide the most up-to-date guidance to you through those interactions.
Thank you.
Can I talk from here?
It's not working.
Can you guys hear me? Okay.
Yes.
Hi, thank you for the very clear presentations. My question is about a new quality attribute dissolution that seems to have been added to the list of tests. And this wasn't something that was required before, and we now see it in the new product-specific guidances. So can you explain your thinking as to why this has been added?
I guess that's a question for me again. Yeah. So yeah, it's depending on the products. If we think the dissolution is relevant to the systemic exposure, we would like to see the dissolution as a, especially when we talk about comparison data, we want to see that comparison data to your existing product to make sure the systemic exposure is going to be comparable. Would that answer your question?
It does. I mean, I guess we were reflecting yesterday with some of our colleagues that for all the other tests, we have very established methods, and this is something completely new. So
right, and it's
an additional burden on.
Yeah, it's not for all the PMDIs. It's only for some of them. If we think the dissolution is relevant in terms of the some of the bioavailability, then we generally would like to see that data.
Thanks.
Thank you for your presentation. It's very interesting. I'm wondering if the agency has any thoughts about what you will allow to be said about flammability on labeling. And I'm sort of particularly concerned about, in the era of misinformation, that somebody who reformulates without using HFAO 1234ZE might choose to label their product as non-flammable to slur anyone who chooses the other propellant.
I'll respond in general, and Craig can add anything. I don't know that we've gotten to the point of thinking about labeling for the flammability issue. I think we would look to the sponsors to propose what the risks are for the MDIs. I know we understand that maybe the manufacturing flammability issues are something you're going to have to deal with, but I don't know at this point if we know how much flammability is an issue for the individual consumer, for the individual MDI. But Craig, maybe you have something to add?
Yeah, I don't think that we would allow statements that would say, like, for example, if you had a product that didn't have something, we don't allow you to say it doesn't have that. So in this case, we probably wouldn't say that this is not flammable. We wouldn't allow that either, so.
Yeah. The thing that motivated my question, and maybe my memory is faulty, is that I thought during the last transition I saw canisters that were labeled CFC3.
Free.
And so I wondered if you...
I think it might have had a timeline, though, where we had them get rid of that after a while, if I remember correctly. But I don't think they have that anymore on there. Well, that's probably because there are no CFCs anymore. But I think it was like it had an end date that after so many years, they wouldn't be able to do that anymore. But
I think we'd be mindful of trying to avoid any promotional language,
yeah.
in terms of fairness.
Hi, this is Elizabeth from the FDA. I think I just wanted to clarify an earlier question on dissolution and why we're asking for it. So you do see our product-specific guidances did include, in certain cases, dissolution being added. And those cases are really focused on when we believe that dissolution is going to be a rate-limiting step to the bioavailability of your API in the lungs. So you only see those in those cases where dissolution is going to be your rate-limiting step. In other cases, you'll see our guidances don't include dissolution. So again, really pay attention to what's in those product-specific guidances of when dissolution is warranted. Another thing we know is dissolution is fairly new for the inhalation space. It has been used for other drug products, and we understand that the development and standardization is still an ongoing challenge. We have done a lot of research on that already, and we do have some initial thinking. So if you have challenges in that, we welcome you to come to FDA to ask those questions and get some more additional guidance if you are struggling in that area. If there's anything else, we definitely welcome any of those type of questions coming our way. Just wanted to provide a little bit of clarity from FDA's perspective on that.
Thanks, Elizabeth. And we are working closely with our generics colleagues since we are citing the PSGs as frameworks for how to think about these programs. So if you have meetings with us, we will often include them in those meetings.
Thank you very much for all the panel members for a great presentation. A lot of learning for us. So my question is actually, since we are, I understand what was required from CFC to HFA, and now we are looking at HFA. And I'm also hearing that there are certain similarities between HFA and LGWP. And based on that, if a company for NDA or ANDA is able to present a robust in vitro and in vivo pharmacokinetic study, would the agency consider alternative BE studies instead of a pharmacodynamic study?
I'll ask Stacy to respond.
So are you indicating an alternative to a comparative clinical endpoint study?
Yes.
I think at this point in the transition, that is what we think we need, in addition to the safety study. As we get more experience, we might consider alternatives, but I would say it's really too early to consider anything other than what we've outlined here today.
Okay, thank you.
Thank you.
Hi,
Rick again.
Yes. So Elizabeth answered the follow-up question that I was about to ask, so I have a new question. In terms, this is a biopharm question. If the reformulated product provides the same input condition within experimental error and so forth of DDU and APSD, what would you expect to be different in terms of an outcome and the value of either an abbreviated clinical study or a further pharmacokinetic analysis, assuming the input condition is the same? What is the rationale for a robust in vivo comparative assessment at that point?
I think that's an excellent question, Rick. We still feel like the linkage between these two important in vitro properties you mentioned with the in vivo performance is still missing. We still need more data and to prove that linkage. I guess that's maybe a brief answer, if that helps. We would attend the idea if the industry, if the sponsor could actually provide the linkage between those, I know most companies will see these are the most important two in vitro properties. However, I think there's still a lack of a comprehensive understanding how those in vitro properties were directly linked to in vivo performance. I think we still need an answer from that in order to, to answer your question, basically.
Thank you. By the way, great presentations, everybody.
Hi, Geraldine Ventoy from Vectura. I didn't hear anyone speak about spacer today, and yet, Steve, you talked about the differences in physicochemical properties on plume geometry, evaporation, timings, and therefore there lies, even with a product with very minimal change in terms of the API being the same, the formulation being essentially the same apart from the propellant, the device being the same in terms of elastomers, valve, and all other aspects, that just merely because of the dipole moment differences and the evaporation differences, that you could get a very similar and bioequivalent product from an in vitro point of view, maybe even from a PK point of view for the product without spacer, but with spacer there would still be differences drawn from the differences in spray geometry that you talked about. So any comment about the qualification for an abbreviated program where you show that similarity in all the things that you've been asking for, but still show differences in spacer, and spacer is in the label, and as we talk about similarities in label includes a recommendation for a name spacer.
Well, I'd say with MDI development generally, we typically don't require or use spacers in our clinical studies or with the in vitro comparisons. Sometimes you'll see some labels that have that in vitro data with spacers, but from our perspective, the introduction of a spacer, and there are so many different spacers, it almost becomes, it's an addition of another device to a combination drug device. And so our requirements are just with the MDI itself. And we realize this is maybe a little different than other regulatory bodies. They do require spacers or allow spacer use in the development programs, but we do not.
I was just going to say, I think your point is well taken. I'll say that I'm in favor of not including requirements for also doing spacer testing with all of the different realistic throat, different flow profiles, dissolution. Yeah, it's going to be really challenging to meet all of those metrics. And so I personally am in favor of not adding that one.
I agree with you, Steve, which is why I bring it up. I think spacer data is very noisy and has been seen to be very noisy also in PK studies where it has been used for some approvals. I think another aspect of the label is how the dose is represented, and there are territorial differences in how you represent dose on labels. As you said, Steve, there are density differences in labels, and therefore in some labels you have the dose presented in from a strength point of view, from a nominal amount of dose point of view, and that will change obviously if you are changing density. So I think at the FDA, it would be good to hear from you as to what you think about that, where you don't necessarily qualify with exactly the same label, though essentially the dose is the same that you're delivering, both in terms of delivered dose and APSD.
I'm not sure we've thought a lot about labeling at this point, to be honest with you. I think if you have a specific situation, that's a discussion we could have. I don't know that we have general feedback at this point.
I think that the reason that we're bringing this up is that obviously we're very pleased to hear about an abbreviated clinical approach, and we understand, that requires us to, as industry, to show similarities or same label. But label covers a lot of things which may or may not be relevant in terms of what the patient receives, and therefore it may be good to get some clarity around that, because, for example, if we think we don't qualify for abbreviated clinical programs, we may not even start the development program to come to you and discuss.
And on the new drug side, the labels don't necessarily have to be exactly the same. There might be differences in the label. I think when we're thinking about carrying over the label, thinking about carrying over indications and labeling claims and not having to repeat those clinical programs in order to do that. So I don't think we were necessarily envisioning that every single word is verbatim the same as the previous label.
My name is Andrew Cooper from Beatrice. Firstly, thank you for presenting such a clear set of presentations this morning. It's given us obviously a lot to discuss in terms of the detail. So I would just like to pick up again on the discussion we've already started here about dissolution. And arguably the point I'm about to make really applies to spray pattern and plume geometry, which have been mentioned as well. And what we're seeing, I think, in this approach that's been presented this morning is to my mind an element of duplication, in the sense that we're seeing dissolution being mentioned for some products, but if really what we're worried about is the resulting in vivo dissolution and exposure, then that's pretty adequately measured, in my view, by pharmacokinetic studies, particularly when you're including the potential for charcoal block studies in some cases, where you're really looking at the systemic exposure from the lung, which is going to be a pretty direct measure of what happens if there's dissolution differences. And so, what's the thinking behind needing both of those things within the data package, when the in vitro test in that case is certainly, at the moment, less well established, less certain in terms of its actual relevance, where you're making a direct in vivo measurement?
I'm not sure. That's a very long question. Try to understand.
I'll give Hailing. I think what you're arguing is that if there's no, why is the in vitro dissolution testing necessary? Because it would really, what we care about most is the PK in vivo exposure.
Correct, yeah. Thank you.
Okay, I think I got it.
Okay.
So another perspective of have a comprehensive in vitro testing in your development program is that also from life cycle management perspective. If you have a comprehensive, complete in vitro understanding and testing and the safe space built around those in vitro testing, then moving forward, if you have some minor changes in your manufacturing processes, minor changes in your formulation, maybe we could use this test as a baseline to approve those post-approval changes. So that's another perspective I want to present here.
That's a very interesting perspective. Thank you.
Hi, I'm Deepika Lakhani. Thank you for an excellent presentation, acknowledging how early we are in this transition in the country. My question is specifically for the long-term safety study requirements that were highlighted in the presentation this morning. Do you anticipate, with the first NDA approval and qualification of the new excipient at a specific dose, that requirement to go away for the following approvals after that?
I'm going to take this. I think it will take more than one approval. In terms of when the safety studies will no longer be required, we don't have an exact time frame or number, but I think one consideration is when the propellant gets added to the inactive ingredient database. That might be one benchmark we're looking for, but I think it will probably take at least several approvals before we get there.
Thank you for the response. Are you anticipating in the 12 weeks of long-term safety between the true propellant systems that we have right now for the LGWP propellant systems, something different or like specific signals to show up, and that's the reason you're requiring those studies to be repeated?
Yeah, I think we don't necessarily know what we will see. I think if lessons from the past tell us anything, it's unlikely there will be novel safety issues with these new propellants, but we don't have any experience with them yet. So I think it's just part of our mission and responsibility just to verify that we think these are safe for long-term use.
Hi,
Thank you for leading with that question because it set me up very nicely for mine. Firstly, thank you for a series of very clear discussion points and for sharing your really thoughts. I'd just like to follow up on the discussion around long-term safety requirements for SARBA relievers. You shared your thinking that a 12-week safety study, perhaps repellent-only study, would be suitable. I guess my concern is that that's not necessarily representative of real-world use and could be an incredibly difficult study to execute, thereby delaying access to these sorts of medicines for patients in the US. I'd be interested if you had explored alternatives, for example, a shorter max daily dose safety study supported by a longer therapeutic dose study, or even supported by real-world evidence if it's available at time of file.
We have not specifically explored any of those options. I think some of this is, our thinking is impacted by some of the proposals we're seeing and what other regulatory bodies are thinking, and trying to hopefully not put too much burden on sponsors, but also get the data we need. I think if a sponsor has a different proposal, we are willing to look at that and entertain that. I think right now, this is sort of what we've landed upon, is what we think is maybe a reasonable duration to get the information we need. But again, we're early, and I think we're willing to discuss alternatives with sponsors.
Great, thank you.
From Viatris. So I understood that at this point of time, very early in the transition, a product on the market which is reformulated with a new propellant is considered a new product because you need to provide a lot of safety data, initial data, and things like that. But in a later course of the development, when you see more and more of these low global warming potential products on the market being registered, and in view of the similarities between the current generation and now this greener propellants, would you consider and revisit your opinion that this would be considered a new product if it's reformulated, an existing one is reformulated with a new propellant, even if there were only a few possibly formulation changes needed?
So I think right now, where we are, like you're saying, with the clinical study requirements, we are saying these submissions should come in as original NDAs.
Right.
At some point in the future, it may be possible to submit supplemental NDAs to support the new propellant product. At what point that happens, we don't know exactly, but I think once we no longer need all the safety data, et cetera, that's probably the inflection point we're looking at. So I think at some point we do anticipate that these could be supplements, prior approval supplements, rather than original NDAs.
Okay, thank you very much.
Does that answer your question?
And I'm very delighted that you all showed the willingness and really encouraging us to come and talk this early and design right program to maintain access of patients to these medicine. Highly appreciate the openness to discuss.
Thank you.
It's Lee from Bestvac. Thank you very much for the presentation and also the panelists. I have two technical questions. The first one regarding formulation. So if we do need to change the formulation slightly, for example, by adding ethanol to the formulation in order to achieve the same performance of the reference product, which does not contain ethanol, would that be considered as a major change to the formulation? And can we still follow the streamlined approach?
Yes, I mean, I think we anticipate that there are going to be some changes in formulation and even maybe some minor changes in the device. And you can still follow the streamlined approach as long as your product quality attributes can line up and the PK. So I think we understand there will be differences for sure.
Thank you. My second question is about the human fact considerations. If, say, we do find that we need to change the shaking procedures and we need to have slightly different priming or repriming schemes, and if those can be justified by properly designed studies, can we still consider this not a critical change and follow the streamlined approach?
So the devil's always in the details, and I would encourage you to come talk to us if that is the case. I think that's the $25 million question here, is exactly how much change is going to be acceptable to still be considered comparable and okay to pursue a streamlined clinical program. I think it will need to be evaluated on a case-by-case basis. I don't think we have like a universal, like, oh, if this is done, this will be okay, if not this. So I think that, I know that's frustrating to hear, but I think that's why we're encouraging frequent interactions with us so we can provide specific guidance on your particular application. So it might be possible, but without knowing the specifics, I don't think we can definitively say that.
Okay, thank you for the answer. The last one, probably a little bit philosophical question. So I'm not arguing or would discuss or agreed about the solution testing, etc. So currently, for the inhaled product, the delivered dose and APSD are main performance criteria in the specification, and which means when we release product, we believe that if you achieve those two criteria to the specification, your in vivo efficacy is guaranteed. Right. On the other hand, I hear that if you achieve those two, we're still not confident that you have a same efficacy. Right. So I know I'm not arguing that you need extra study to support, but that's just my. Philosophically, there's some contradictory question there.
Yeah, I guess we can step back, take a step back to look at from the more big perspective, right? When you use these two important properties to release your product is called QBD, quality by design.
Yeah.
It's built in your quality design, in your formulation, in your device, in your manufacturing and manufacturing process control. Now, you are dealing with a new product, with a propellant, with some maybe changes in your formulation, and with also maybe minor changes in your device. Then we have to think maybe a little bit differently. So this is maybe another reminder. I know PSG be mentioned in many of our talks, but if you look at our PSG, the prerequisition is Q1, Q2, and the device similarity. So I also want to remind all the new drug sponsors, when you consult PSG, please take that into consideration to see how the risk associated with your non-Q1, Q2 formulation device change will be mitigated by additional data. I hope that answers your question.
I think I answered my question. So this is, for example, the solution more relating to Q3, for example. If
Right.
the first line changes that maybe. But that's why I say that. I'm not arguing that, but I just want to have a more clarification. Thank you very much.
Right. I think from broad scientific perspective, the linkage between these two important physical properties to directly link to in vivo performance is still lacking. I think we still need more evidence to show that, as I mentioned in my response to Rick's question.
Yeah, thank you.
I don't know if we want to take any online questions. I know there's like a lot of people online I don't know what the organizers want us to do. Yeah, let's take a few online questions since, sure. While Banu's coming up here, maybe we can do one more question in the room.
Oh, okay. Yeah, I first of all, thank you for this morning, excellent session this morning. A lot of the presentation I heard the first time. Okay, I just want to get a quick clarification regarding the word stepwise used in Aishah, your presentation. When you see stepwise, are you referring to, say, we're talking about two clinical programs. One is streamlined, the other one is a full clinical package, right? So when you talk about stepwise, are you referring to, say, if you failed the first program, then you go to the second program?
Well, I think what we're talking about is if you want a streamlined approach to your clinical program, I don't think we can agree to that up front. We need to see the product quality data and then the PK data before committing that, okay, you don't need a full clinical development program. So it's sort of risky on this, a sponsor to sort of plan on a streamlined development program, but not know whether that's going to be acceptable or not. So from our perspective, it's just showing us some data along the way to make sure that we're all on the same page and that we agree a streamlined program is okay. Because otherwise, you might have to shoot off and do more clinical studies when you think you just need a comparative clinical endpoint study.
Thank you for the clarification. The reason I'm raising up here is, there is Europeans using a so-called stepwise approach for their bioequivalence assessment of their drug product, which is different from what you're talking here, which is a stepwise procedure for the acceptance of these two programs. So I just want to make this clarification here so that people understand when we talk about stepwise, it's not what, the stepwise mentioned in the European guidance.
Okay.
Thank you.
Thank you. There's a lot of loaded terminology.
There are a lot of questions online, and I'm going to try to filter through some of them to also give folks who have questions in the room still a chance to speak. There were a number of questions for Steven Stein in the beginning that came through, so I want to make sure I get to some of them. So one of them is, do you see filling issues with 152A since it is more flammable?
Yeah, that's definitely been one of the things that's been discussed, and there will need to be some modifications, but it's not an insurmountable task to modify the systems. But yeah, it depends on probably case by case what a company's system, exactly how it's set up, but probably some modification needed.
I'll thank you on behalf of the asker. And then another question for Steven Stein. So inhalation products can be very sensitive to throat anatomy. Do you think it's especially important to evaluate this factor in the aerosol lab continuously during the development of these type of products?
I think if you look at some of the conversations and presentations that are going around, I would highly recommend considering that, as I think it will be expected. So yeah, I think it would be a good thing to do.
Perfect. Thank you so much. And then finally, another question. Can you comment on the impact of actuator design attributes other than jet length that can be potentially used to manipulate spray pattern?
Well, jet length, for spray pattern, jet length is definitely an important parameter. My experience is orifice diameter doesn't have a huge impact on spray pattern, but it could provide some. Then you've got the cone angle, various things you could evaluate. And there's some literature. Hugh Smythe a while back did a publication that could be of relevance. Now it's obviously for older propellants, but there's some literature that could be useful.
Great, thank you. There was also a question online about the clinical study designs, and so I'm going to direct this to the clinical team. The question was just asking on an elaboration on why the use of a placebo arm for assay sensitivity.
Yeah, I think it's to have assay sensitivity in the test and make sure we're not missing anything. It's also included in many of the comparative clinical endpoint studies and the PSGs, and I think we're carrying that over in some ways, but just to make sure that the test is sensitive to change.
Perfect. And there was a couple of questions about charcoal blocks, so I will direct these to Sneha. As per literature, if the Tmax is less than 5 minutes and AUC 30 is measured, can the charcoal block PK studies be waived off?
I cannot comment on behalf of a hypothetical scenario. So my recommendation would be that you come talk to us about the challenges you're experiencing, and we may be able to guide you better.
And there was also a more specific question about how to select the dose of charcoal. And does it need to be essentially the US FDA approved or a US sourced charcoal product? Is that something you can comment on?
So I think there's a lot of literature on this, and I would ask that sponsors look at the literature on this before coming to us and talking about it. But yes, the dose of charcoal, we need to have it justified, and it should be enough to block almost all of the GI absorption.
Great. Okay, I'm going to go back to people in the room, and then if there's time, so we can alternate back and forth.
Thank you. I am a repeat offender, Sally.
I know this.
One quick question and then a more lengthy question for Luke from the FDA. My understanding is that except for injections, ophthalmics, and otics, that's Q1, Q2 is not required, even though it may be recommended. Does that apply to PSGs?
Right. Most PSG will have two options. One is for Q1, Q2. One is for non-Q1, Q2 formulation, but we also have that language, similarity device. So that's kind of a general advice, when you consult, keep that in mind. When you take the option one in the PSG, the Q1, Q2, if you can justify that why the formulation difference, still it's an option right there.
I just wanted that clarified. Thank you.
Yeah, absolutely. Thank you for that.
Yeah. Luke from the FDA, got a question for you. My understanding is that the EPA, is working to help research in this area of transition of propellants, and that is some other sectors that they're able to support that. Is there any mechanism by which the academics in this room could get, for example, some research support to address some of the questions that have been asked to Stephen that may not appear in the literature yet, such as the behavior of mixture of propellants, which I asked about because it may be a way for applicants to get around patent issues that use one single propellant. Things like that. Is there any mechanism for that to which either companies or academics can get involved with?
Sure. So just to make sure I understand the question, are there research grants or other opportunities to support trend?
Thank you for summarizing that simply, yeah.
Thank you. Just wanted to make sure. So I know in my specific group, we don't have any research sort of budget that way. We're usually reviewing what companies submit as an alternative and reviewing their research. But having said that, the agency, EPA, does often have research grants across a number of topics for environmental projects. I'm not aware of any that are specifically targeted for this use, but doesn't mean that there might not be something out there. So maybe we could follow up after, and I can try to do a little digging just to see what requests are out currently or if there's anything planned.
Thank you very much.
So it looks like the Richards are ganging up on Luke. So at recent respiratory drug delivery meetings, John Pritchard has kind of suggested that the shortage of HFAs will bite more quickly than perhaps your timeline suggested. So thinking about your allocation system, I'm just wondering, does it in any way give priority to companies that avoid wasting HFAs during their filling processes, meaning that not everything they buy ends up in a canister that's then used by a patient?
Yeah, it's a good question. So in terms of how the program is structured now, it doesn't. It does base it on how much are you using, how much are you purchasing in a given period of time. We do gather information on the disposition of what you have, whether you're destroying certain amounts at the end of life, again, so it's not being emitted, or if you have an off-spec batch or something like that. But it's not something that we currently do.
I just thought a relatively easy bit of data to collect would be how much a company is allocated and how many cans they put on the market would be a simple metric that you could judge efficiency.
Thanks.
Thank you for your great presentations. I have a question regarding to the streamlined development approach.
So in a more recent update for a batch of PSGs, and taking Albiro as an example, and there are two options provided for development. Option one has the precondition of Q1, Q2 sameness, and it allows to do two more in vitro alternative studies, and in lieu, plus additional alternative B studies in lieu of the comparative clinical endpoint study. And option two is still the traditional approach with the Q1, Q2 not to be required the same. So my question is for the new GWP program, if we're aiming for the streamlined approach, and would that we have to follow the option two because the propellant is Q1 qualitative indifferent? So would that mean we have to follow the option two for development if we wanted to follow the streamlined approach as a reference? Thank you.
I mean, so for new drugs, we're using the PSG sort of as a framework, but that's not necessarily a requirement. It's to give sponsors something to reference for details about our general thinking about the data we want to see and the study designs. I would say for an individual program, you should talk to us. Right now, we expect to see a comparative clinical endpoint study. So it would be the second option. But some of our advice is not going to follow exactly the PSG. We're using it sort of as a guide, a reference, but not necessarily a recipe, like you do this, and it doesn't necessarily apply to the new drug side.
Okay, got it. Thank you.
Hi, Elizabeth. Maybe I just can clarify a couple points on that as well. I will be presenting tomorrow a little bit more about the PSGs and how they apply, and especially regarding the formulation sameness for option one. So there may be some situations where you can justify it through option one. I'll get into detail about that tomorrow, so please hang on to your hats till we kind of cover that tomorrow. And I did hear some comments about DDU and APSD being our main comparator studies that we think are very important in the in vitro setting. I highly encourage you guys to participate in our small working groups discussions where we're really going to try to tease out what are going to be the important tests that we want to use to establish comparability between HFA products and the LGWP. So please provide your thoughts there, and hopefully we can have some more discussion on certain studies in that realm. So thanks.
I wanted to go back to the question previously to the EPA. And it's quite a controversial question. We are looking in Europe, I'm more familiar with the F-gas situation, I'm less familiar in the US with AIM and allocations. But my understanding is that this applies to manufacturers and importers of HFCs. By the virtue of what Richard said before, manufacturers, and there's good quality manufacturers existing outside of these territories in India, for example, and a growing number of doses, MDI doses in the US coming from manufacturing in India. By not looking at it from a global perspective, are we now actually preferring product to be manufactured elsewhere, or looking at product to be manufactured elsewhere that don't fall to the same extent under the allocations of the EPA and the Europeans? How do we, and perhaps the new administration will have some say around this, but how do we actually get manufacturing back into the US if it's still affordable in the US to do so?
And just to clarify, this is sort of if you're manufacturing in the United States, you need the allowance to, get the chemical. Whereas if you're importing the finished, MDI, there's no allocation needed, right? That's the question, yeah. So I think, this is something that did come up when we structured, established the original framework rule for this program. And what we've generally seen is because of that global program to phase down, that sort of puts the incentives generally in the right direction to keep, a reasonably level playing field. Having said that, obviously there are other folks that have different views on, how we should handle it. But in terms of how the structure is currently set up, yeah, it does have a difference. But having said that, right, the application-specific allowance program, which prioritizes access to HFCs, priority access to HFCs for someone who does need to manufacture here in the US, somewhat under, sort of cuts against that, the sort of a bit of the premise of your comment, because again, we are allocating based on need for that entity. So, again, other countries have different regulatory regimes, and they're, the manufacturers there are having to comply with those requirements as well. Obviously, they're very different in certain places, but at least for us, for the current setup, there is that priority access, so it's less of an issue. Thanks.
Yeah, I have a question, but before I ask that question, I have a comment about what Elizabeth just said. So tomorrow when you give your talk, Elizabeth, can you please cover both Q1 differences as well as Q2 differences because of density differences?
Sure, I can elaborate a little bit about that density differences and what that may mean for Q1, Q2 sameness.
Yes. Okay, thanks.
You're welcome.
So the question that I have is, under what circumstances do you think that when we switch to MDIs with a low GWP propellant, we may need a specific patient handling study?
I think that's where you have to do the use-related risk assessment and go through that process, and that's when you will determine if you need to have a human factor study or not. Is that what you're asking?
Yes.
Yeah. So you perform a use-related risk assessment, and I think there's a guidance on that or standard guidance about how to do that assessment.
Yeah, there are a couple of ways that you can do it. One is through that guidance document. The other is when in the lab you do patient-simulated use testing to try and replicate how a patient would use the product.
I'm not sure that's part of the standard assessment, so it could be supplemental if you want to provide that information, but I think it's sort of a standard assessment to compare the products and the differences and whether there's going to be an impact on the user interface and the patient experience.
Okay, thanks.
Couple more questions from online?
Sure.
I know we're at noon, but there's a couple of questions. Can you speak to the possibility of leveraging in silico modeling to further streamline the transition process, specifically in relation to PK and PD considerations?
I think I'm going to have to ask our OGD colleagues to weigh in on that, because from the NDA side, we don't have experience with the in silico modeling.
Yeah, hi, Ross Wallinga, FDA. This is really a question for new drugs as far as what you're willing to accept, but from generics point of view, we look for modeling to perhaps inform on review standards. For example, if you're thinking about your particle size distribution testing and you're having trouble passing, you might be able to use modeling to show an in vitro and vivo correlation such that you can demonstrate that there may be allowable differences that are not really the current review standard. And we can also consider in silico models as a additional study if you're doing something like an alternative bioequivalence approach and you want to leave out certain studies that are in our guidances. So from generics, that's how we look at it, but as far as new drugs, I think that would depend on how you want to view that.
I think you would probably need to come in with your proposal on how it fits into your overall program and your specific situation. Like I said, we're working with our OGD colleagues on this because, as you can see, we're leveraging or using a lot of the knowledge they've gained and have put out in the PSGs. So thanks, Ross.
Sure.
Maybe, Sally, I can make it. So in terms of the role of in silico model to bridging the new formulation to the existing formulation, I think one reminder is that this has to be looked at in the general bridging picture. Could be useful to provide a supportive evidence, especially to link the in vitro characterizations to the in vivo performances. However, we want to remind this those models has to be looked at very carefully in terms of validation, and especially like for this purpose, sensitivity analysis. And I think circle back to my original kind of thoughts is that has to be looked at in the big picture, how this link to, what other evidence you have and what kind of consequence it would have if we use the model to make the decision.
Okay, I think you might have the last question. No pressure.
All right. This is a clarification question. Ann Purrington from Kendeva. With respect to the human factors topics that were discussed in the framework proposed, the mention of use-related risk analysis and then the 3 components were included. And on a terminology basis, I just wanted to have clarity around, is that referring to the comparative threshold analysis guidance where you're trying to demonstrate on those 3 components that there are no differences and therefore the device user interface is comparable, rather than the use-related risk analysis approach?
We're not the human factors experts here, so we may not have the right answer for you unless anyone on the panel knows for sure.
Okay, later,
Sorry, Ann.
Later.
Okay, we'll see. We might see what we can do on that. Oh, wait, Elizabeth, you have
Hi,
the
Ann.
answer?
Maybe I can provide a little bit of clarity. I think it depends if you're comparing to your old product versus your new product. I think you can use the comparative analyses as a way to do that. But if you're developing an entirely new product, then your URRA is going to be part of your human factors, right? So I think it just depends on the context. And if you need more clarity, I'd suggest coming to the agency for discussion and probably get a little more specific guidance from our human factors experts.
Okay, I think that brings us to an end session 1, and is there anything from the organizers about instructions for lunch or that way? Thank you.